Hello there, and welcome to Life Beyond Seen, where the familiar world has a way of quietly hiding its deepest secrets just beneath the surface, and where the things we have never seen are often closer than we think. Tonight, we're going somewhere that most people have never heard of, even though it sits in one of the most visited seas on the planet. Not to the edge of some far polar ice sheet, not to the remote Pacific, not to some distant corner of the globe that requires 3 connecting flights and a boat ride. We're going to the Caribbean, or rather, we're going beneath it. Because the Caribbean that most people know is a surface world. It is the world of warm turquoise water and white sand, of cruise ships and snorkeling tours, of afternoon rain showers that pass quickly and leave the air smelling clean. It is a sea that has been photographed from every angle, mapped by satellites, crossed by container ships and fishing boats and pleasure yachts for hundreds of years. People know what the Caribbean looks like. They have posters of it on their walls, and yet beneath all of that familiar warmth, beneath the postcard colours and the gentle waves, there is a place that almost no human being has ever seen. A place where the seafloor drops away into a darkness so complete and a pressure so immense that even our machines struggle to reach it. A place that holds, in all likelihood, creatures that have never been named, never been photographed, never been observed by any eye, human or mechanical. That place is the Cayman Trough. It sits in the Western Caribbean Sea, tucked between the southern coast of Cuba and the northern coast of Jamaica. On a map, it does not look dramatic. It is just a long, thin feature running roughly east to west, stretching approximately 1,400 kilometers in length, but only about 80 kilometers wide in places, not particularly broad, easy to overlook. But what the map does not convey is what is happening beneath the seafloor surface of that narrow strip of ocean, because the Cayman Trough drops. It drops in a way that is genuinely difficult to picture. The deepest measured point sits at approximately 7,686 meters below sea level. That is just over 7.6 kilometers of water, stacked straight down, cold and dark and pressing. To put that in some kind of human scale, if you placed Mount Everest at the bottom of the Cayman Trough, its peak would still be more than 2,700 meters underwater. The summit of the highest mountain on Earth would not come close to breaking the surface. That makes the Cayman Trough the deepest point in the entire Caribbean Sea, and it is also one of the deepest trenches in the whole Atlantic basin, a fact that tends to surprise people who think of the Atlantic as shallower and quieter than the Pacific. It is not, and the Cayman Trough is one of the clearest reminders of that. Now I want to pause here for just a moment, because before we go any further into this place, I would genuinely like to know where you are listening from tonight. What part of the world has you? Is there a window nearby? And if so, what is the temperature on the other side of it? Are you somewhere warm, where the idea of cold dark water 7 kilometers below the surface feels very far away? Or somewhere cold, where the thought of that darkness feels almost comfortable? Wherever you are, whatever the hour is, I am glad you are here. This is the kind of story that asks for a quiet room and a little patience, and I think it rewards both. If you find yourself enjoying this kind of slow, careful exploration, I would be grateful if you considered liking this video and subscribing to Life Beyond Seen. It is a small thing, but it genuinely helps this channel reach other people who might be looking for exactly this kind of company at the end of a long day. There is no pressure, of course. Just know that it means a great deal now. Back to the Cayman Trough. What makes this place so quietly remarkable is not just its depth, though the depth alone is extraordinary. It is the contrast between what sits above it and what lies below. Above the trough, the Caribbean does what the Caribbean does. Sunlight filters down through warm, clear water. Sea turtles move through coral gardens. Cruise ships pass overhead carrying thousands of people who are, at that exact moment, probably thinking about what to order for dinner. The surface world here is known. It has been documented, catalogued, visited, and revisited. There are dive guides and tourism maps and marine protected areas. The shallow Caribbean has been studied quite thoroughly as these things go. But a few kilometres directly beneath all of that ordinary activity, the world becomes something else entirely. The light disappears. The temperature drops toward freezing. The pressure builds to levels that would crush an unprotected human body in an instant. The seafloor is not white sand and coral. It is dark volcanic rock, fractured and active, shaped by forces that are still moving right now, tonight, while you are listening. And in the deepest parts of the trough, in the places where the seafloor has never been visited by any camera or any vehicle, there is simply no data, no images, no species lists, no samples, just depth and darkness and whatever is living there. That absence of data is not a small thing. It is, in a way, the whole story. The Cayman Trough is not a relic. It is not a passive feature of the seafloor that simply happened millions of years ago and has been sitting quietly ever since. It is geologically alive. It is being actively shaped by forces deep within the earth right now, in ways that create conditions unlike almost anywhere else on the planet. And those conditions, as it turns out, are exactly the kind that life tends to find interesting. The surface world of the Caribbean is mapped. It is known. It has been photographed from orbit and from the deck of a boat and from the mask of a snorkeler. But the Cayman Trough just below is something else. It is a place where the Earth is still making itself, and that is where the story of what might be living there, unseen and unvisited, actually begins. Let us start with the geography in a little more detail, because it helps to have a clear picture of this place before we descend into it. The trough runs roughly east to west, as mentioned, beginning near the Cayman Islands in the west, and extending toward Jamaica and Hispaniola in the east. It is bounded on its northern side by the Cayman Ridge and on its southern side by the Nicaraguan Rise. These are underwater geological structures, old and massive, that frame the trough on either side like the walls of a very long, very deep corridor. The Cayman Islands themselves, which sit near the western end of the trough, are actually the tops of underwater mountains. The islands you can stand on and look at with their beaches and their diving resorts and their famous 7-mile beach are just the peaks of a submerged ridge poking above sea level. The actual depth of the water around them, if you go in the right direction, drops away very quickly. A few kilometres from shore you can be in water thousands of metres deep. The islands give no hint of this. They look like islands, but the seafloor beneath the surrounding ocean is doing something extraordinary. Now the trough is long and narrow and very deep, but it is not uniform. The deepest section, around 7,686 metres, is in the western portion. As you move east, the depth changes. The terrain is complex, with ridges and valleys and scarps and sediment plains all mixed together. This is important because it means that even within the trough, different zones exist at different depths, under different pressures, with different geological characteristics, and as we'll explore, different zones tend to host different kinds of life. The deepest point has a name. It is called the Bartlett Deep, sometimes also referred to as the Cayman Deep. It was first measured by sonar surveys, and its depth has been refined over time as measurement technology improved. 7,686 metres. That number is worth sitting with for a moment. It is not an abstraction. It is a real physical distance. If you could stand at the bottom of the Bartlett Deep and look up, the surface of the sea would be more than 7.5 kilometres above your head. The sky would be invisible. The sun would be invisible. The entire world of light and warmth and familiar life would be impossibly far away. And yet life is there. We know that much. We do not know what all of it is, but we know it is there, because in the places we have managed to reach, we have found it. Extraordinary, improbable, beautiful life, surviving in conditions that should, by any ordinary intuition, make survival impossible. The contrast between the warm, familiar Caribbean above and the cold, pressurized darkness below is not just poetic, it is scientifically significant. It means that the Cayman Trough has been developing its own biological world in relative isolation, cut off from the surface by kilometres of water, cut off from other deep environments by geography and distance, doing its own thing in the dark for a very long time. What exactly that thing is, in its fullness, we do not yet know. What we do know is that the trough is not just a hole in the seafloor. It is an active geological system with its own internal heat sources, its own chemistry, its own currents and pressures. It is a world within a world, and the first key to understanding why it might harbour creatures no human has ever seen lies in understanding what is happening geologically at its heart, in the slow, dark spreading of the seafloor that sits at the centre of everything. Because the Cayman Trough is not just deep, it is alive in the geological sense, and that is where things start to get genuinely interesting. The forces that built this trench are still at work, the seafloor is still moving, new rock is still being made, and the heat that drives all of that is doing something to the water above it, something that has consequences for every living thing in the area. Understanding those forces is the next step into this story, and it changes everything about how we think about what might be down there, waiting in the dark. To understand why the Cayman Trough is the way it is, you have to go back to the plates. Not the tectonic plates as an abstraction, not the kind of vague gesture toward a continental drift that gets mentioned once in a classroom and then forgotten, but the actual physical reality of two enormous slabs of rock grinding and pulling against each other at the bottom of the Caribbean Sea. Because what is happening in the Cayman Trough is not quite what happens in most deep ocean trenches, and that difference matters enormously. Most of the world's deep trenches form through subduction. That is the process where one tectonic plate slides beneath another, diving down into the mantle, dragging seafloor with it, and creating a long, narrow depression at the boundary. The Mariana Trench works this way. The Japan Trench works this way. Subduction trenches are relatively well understood, at least in their basic mechanics. One plate goes under, one plate rides over, and the trench forms at the line where they meet. It is a violent, compressive process, and the trenches it produces are enormous. The Cayman Trough is different. It did not form through compression, it formed through extension, through pulling apart. The North American plate and the Caribbean plate are not colliding here in the traditional sense. They are sliding past each other laterally, in opposite directions, along a transform fault system. The North American plate is moving roughly westward relative to the Caribbean plate, and as these two massive slabs grind past each other, the boundary between them is not a clean line. It is a complicated zone of faults and fractures, and in certain places along that boundary, the motion has created gaps, spaces where the crust has been pulled sideways, stretched thin, and in some places pulled apart entirely. The Cayman Trough is one of those spaces. Geologists call it a pull-apart basin, and it is exactly what it sounds like. A place where the earth has been stretched open. The key structures that define the trough's boundaries are the Oriente Fault, which runs along the northern edge, and the Swan Islands Fault Zone, which runs along the southern edge. These are not small features. The Oriente Fault in particular is a major transform fault, comparable in some ways to the San Andreas Fault in California, though less studied and far less visible to the surface world. These faults are the walls of the trough in a geological sense. They are the boundaries along which the lateral motion of the plates is accommodated, and between them, the crust has been stretched and thinned and pulled downward into the deep basin that we now call the Cayman Trough. What this means in practical terms is that the trough has a different character from subduction trenches. The seafloor inside it is not old, compressed oceanic crust being dragged toward the mantle. It is relatively young, thin crust that has been pulled apart and is being actively replaced. And that replacement is happening through a process that makes the Cayman Trough one of the most geologically unusual places on Earth. Sitting near the centre of the trough, at a depth of around 5,000 metres, is the Mid-Cayman Spreading Centre. This is a mid-ocean ridge, the kind of geological feature more commonly associated with the middle of the Atlantic or the floor of the Pacific, but here it is hidden inside a narrow enclosed basin in the Caribbean. A spreading ridge is a place where two plates are moving apart and new seafloor is being created to fill the gap. Magma wells up from the mantle, cools, solidifies, and becomes new oceanic crust. The process is continuous, slow, and ancient, and it is happening right now at the bottom of the Cayman Trough. What makes the Mid-Cayman spreading centre particularly unusual, even among spreading ridges, is its combination of depth and spreading rate. Most mid-ocean ridges sit at depths of 2,000 to 3,000 metres. The Mid-Cayman ridge sits at around 5,000 metres, making it one of the deepest spreading ridges on the planet. And it spreads slowly, very slowly. The rate of spreading at the Mid-Cayman Centre is roughly 15 millimetres per year. That is about the rate at which a fingernail grows. By comparison, the East Pacific Rise, one of the fastest spreading ridges in the world, moves at up to 150 millimetres per year. The Mid-Cayman Ridge is at the opposite end of that spectrum, creeping apart with a patience that makes geological time feel very real. Slow spreading matters for more than just the pace of seafloor creation. It also affects the structure of the ridge itself and the kind of geological activity that happens along it. Fast-spreading ridges tend to produce relatively smooth, flat seafloor with a consistent volcanic character. Slow-spreading ridges are more rugged, more structurally complex, and they tend to expose more of the deep mantle rock called peridotite at the seafloor. When seawater reacts with peridotite, a chemical process called serpentinization occurs, and that process releases heat and produces hydrogen. Hydrogen is a chemical energy source, and chemical energy sources in the right context can support life. This is where the geology of the Cayman Trough stops being purely academic and starts being directly relevant to the question of what might be living there. Because the combination of a deep, slow-spreading ridge with active magmatic heat and serpentinization reactions creates conditions that are, in a very specific way, ideal for hydrothermal vent systems. And hydrothermal vents, as scientists have discovered over the past few decades, are among the most productive and biologically extraordinary environments on Earth. A hydrothermal vent, at its most basic, is a place where seawater seeps down into the seafloor through cracks in the crust, gets heated by the rock below, and then rises back up through the seafloor carrying dissolved minerals. The temperatures involved can be extreme. At some vents, the fluid emerging from the seafloor exceeds 400 degrees Celsius. That is well above the boiling point of water at the surface, but at the pressures found at depth, the water remains liquid. When this superheated, mineral-rich fluid meets the cold seawater above it, the dissolved minerals precipitate out rapidly, forming solid structures. These structures grow upward from the seafloor like chimneys, sometimes reaching several meters in height. The ones that release dark, mineral-heavy fluid are called black smokers. The ones that produce lighter, more diffuse flow are called white smokers. Both are remarkable to look at, if you have the means to look at them. What makes hydrothermal vents biologically significant is not just the heat or the minerals, it is the chemical energy. The fluid coming out of the vents is rich in compounds like hydrogen sulfide, which certain microorganisms can oxidize to produce energy. This process, called chemosynthesis, is the foundation of an entire food web that has no connection to sunlight. At the surface of the ocean, photosynthesis drives almost everything. Plants and algae capture sunlight and convert it to organic matter, and everything else builds on that. But at a hydrothermal vent, sunlight is irrelevant. The energy comes from the chemistry of the Earth itself, from the heat and the minerals and the reactions happening in the rock. Bacteria that perform chemosynthesis form dense mats on the vent structures and in the surrounding sediment. Larger organisms, shrimp, crabs, snails, worms, feed on those bacteria or on each other. An entire ecosystem runs on geological energy, completely independent of the sun. The existence of this kind of life was not known until 1977, when the submersible Alvin dove to hydrothermal vents on the Galapagos Rift and found communities of organisms so dense and so unexpected that the scientists on board were genuinely astonished. Tube worms 2 meters long, clams the size of dinner plates, shrimp in swarms so thick they covered the seafloor. None of it had been predicted, none of it had been imagined in quite that way. The discovery changed how biologists think about the conditions necessary for life, and it opened a new set of questions about where else on Earth, or beyond it, similar ecosystems might exist. The Cayman Trough has its own hydrothermal systems, and they are in some ways even more extreme than those first vents at the Galapagos Rift. But we will come to that in a moment. For now, what matters is understanding the geological architecture that makes them possible. The Mid-Cayman Spreading Centre, sitting at 5,000 metres depth, with its slow spreading rate and its exposure of deep mantle rock, creates exactly the conditions that drive vent activity. Magmatic heat from below warms the circulating seawater. Serpentinization reactions in the peridotite produce additional heat and hydrogen. The result is a hydrothermal system that is not just active but chemically rich in ways that go beyond what you find at faster spreading ridges. There is also a structural consequence of the slow spreading and the pull-apart geometry of the trough. Because the basin has been formed by lateral extension rather than by subduction, the seafloor inside it has a particular topography. It is not a simple, smooth incline from the edge down to the deepest point. It is broken and complex. There are fault scarps, steep walls where blocks of crust have dropped relative to each other. There are elevated areas, underwater mountains and ridges, where the geology has pushed material upward. There are sediment-filled plains in the deeper sections, where fine material has drifted down from above and settled over millions of years. And there are the vent fields themselves, sitting on this rugged terrain at different depths, each with its own chemistry and its own biological community. The narrowness of the trough adds another layer of complexity. At only about 80 kilometres wide in places, the trough is not a broad open basin. It is more like a slot, a long narrow corridor of extreme depth bounded by steep walls on either side. This geometry affects how water circulates inside it, how sediment accumulates, and how isolated the biological communities within it are from the surrounding ocean. Organisms living in the trough are not just separated from the surface by depth. They are also separated laterally by the walls of the basin and by the cold, deep water that fills the space between the vent fields and the broader Caribbean. This isolation, both vertical and horizontal, is one of the key reasons why the trough may harbour life that is genuinely different from what has been found elsewhere. The tectonic forces that created the trough are also still operating. This is not a fossil landscape. The North American and Caribbean plates are still moving relative to each other. The faults are still active. The spreading centre is still spreading. Earthquakes occur in and around the trough with some regularity, a reminder that the geological processes at work here are ongoing and energetic. In 2010, a magnitude 7.1 earthquake struck near Haiti, not far from the eastern end of the trough. The seismicity of the region is a direct consequence of the same tectonic setting that created the basin. The plates are not finished moving. The trough is not finished forming. That ongoing geological activity has a biological implication that is easy to overlook, because the seafloor is still being made, because new rock is still being exposed to seawater, because the hydrothermal systems are still active and still evolving. The environment that organisms living in the trough experience is not static. It changes. Vents can go dormant as fluid pathways shift. New vents can open as cracks form in fresh sea floor. The chemistry of the fluid can change as the underlying rock changes. Organisms living in this environment have to be adaptable, or they have to find the new vents when the old ones cool. This pressure toward adaptability, combined with the isolation of the trough, creates conditions that are genuinely favourable for evolutionary novelty. Species that live here are not just surviving in an extreme environment. They are surviving in an extreme environment that keeps changing, and that requires a particular kind of biological flexibility. All of this, the pull-apart basin, the slow-spreading ridge, the serpentinization reactions, the hydrothermal chemistry, the rugged and complex terrain, the ongoing tectonic activity, adds up to a geological setting that is, by any reasonable measure, extraordinary. Most deep ocean trenches are simpler than this. They are deep, and they are cold, and they are dark, but they do not have an active spreading ridge at their centre, producing new seafloor and driving hydrothermal systems. The Cayman Trough has all of that, compressed into a relatively narrow basin in the middle of a sea that most people associate with sunshine and clear blue water. Understanding this geology is not just background information. It is the explanation for why the Trough's biological communities are so unusual, and why the deeper, unexplored sections of the Trough might hold organisms that have not been catalogued anywhere on Earth. The geological conditions set the stage. The biology follows from them, and the biology, as it turns out, is where things become genuinely astonishing. Because sitting at the bottom of this geologically active, hydrothermally heated, tectonically restless basin are vent fields that were not even known to science until 2010. And what scientists found when they finally reached them was not what anyone had expected, even given everything we already knew about hydrothermal ecosystems. The creatures living at the Cayman vents were not simply the same organisms found at other Atlantic vent fields. They were something different, something that had been evolving in this specific place, under these specific conditions for a very long time. And if that is what scientists found on their first brief expensive visit to the vents they could reach, it raises an obvious and quietly thrilling question about the parts of the Trough they could not. The first time scientists actually laid eyes on the BB vent field, they were watching through a camera mounted on a remotely operated vehicle descending through nearly 5 kilometers of dark water. The year was 2010. The ship above was the RRS James Cook, operated by the National Oceanography Centre in Southampton, and the expedition had been planned specifically to search for hydrothermal activity in the Cayman Trough. They had good reason to believe the vents were there. The geology all but demanded it. But believing something exists and actually seeing it for the first time are two very different experiences, and the footage from that first descent into the BB field is, by any measure, remarkable. What the camera showed was a field of hydrothermal chimneys rising from the seafloor, venting superheated fluid into the surrounding water. The temperatures measured at the BB vents exceeded 400 degrees Celsius. To put that in some kind of everyday perspective, water at sea level boils at 100 degrees. The fluid coming out of these chimneys was more than 4 times hotter than boiling water, and yet it did not flash into steam, because at that depth the pressure is so immense that the water remains in a supercritical state, somewhere between liquid and gas, behaving in ways that do not follow the rules we're used to at the surface. It exits the vents as a shimmering, turbulent plume, carrying dissolved minerals that precipitate almost instantly when they contact the surrounding cold seawater, building the chimney structures upward over time in dark, fragile columns of sulfide minerals. These are what oceanographers call black smokers, and the BB field contains some of the most extreme examples ever documented. At approximately 4,960 meters below the surface, the BB vent field holds the distinction of being the deepest known hydrothermal vent system on Earth. That record, as of the most recent confirmed data, still stands. The depth matters not just as a number, but as a physical reality. At that depth, the pressure is crushing. The temperature of the ambient water away from the vents is just a few degrees above freezing, and the darkness is total and permanent. No sunlight has ever reached this place. No photon from the sun has touched these chimneys or the creatures living on them. Whatever happens here happens entirely on the energy budget of the Earth's interior. Discovered during the same 2010 expedition, the Vondam vent field sits at a considerably shallower depth of around 2,300 meters on the flanks of an underwater mountain called the Mount Dent Oceanic Core Complex. The two vent fields are not far apart in terms of horizontal distance, but they are separated by more than 2.5 kilometers of vertical ocean, and they are chemically and physically quite different from each other. Where BB is characterized by high-temperature black smoker chimneys, Von Dam produces what is described as diffuse flow, lower temperature fluid seeping more gently through the seafloor across a broader area. The Von Dam fluids are rich in methane and hydrogen, products of the serpentinization reactions happening in the underlying peridotite rock. The chemistry is different, the temperature is different, and as a result the biological communities at the two fields are different as well, even though they exist within the same trough, shaped by the same tectonic forces. To understand why any creature can survive in either of these places, it helps to step back and think about how hydrothermal vents work at a basic level. Cold seawater percolates down through cracks in the seafloor, sometimes descending several kilometers into the crust. As it moves deeper, it is heated by the magmatic or geothermal energy below. Along the way, it reacts with the surrounding rock, dissolving minerals and becoming chemically transformed. Eventually, this hot, mineral-laden fluid finds a path back up through the crust and exits through the seafloor at the vent site. When it hits the cold ambient seawater, the sudden change in temperature and chemistry causes dissolved minerals to precipitate out, forming the chimney structures that grow upward over time. Some of these chimneys can reach heights of tens of meters. They are fragile, built from minerals that can crumble if disturbed, but they are also constantly being rebuilt as long as the fluid keeps flowing. The key biological insight is this: the fluid exiting the vents is rich in hydrogen sulfide, methane, and other reduced chemical compounds. Certain microorganisms, bacteria, and archaea have evolved the ability to oxidize these compounds to extract energy, in much the same way that plants use sunlight to drive photosynthesis. This process is called chemosynthesis, and it is the foundation of the entire vent ecosystem. These microorganisms form dense mats on the surfaces of chimneys and surrounding rock. They colonize the gills and bodies of larger animals. They exist as free-living cells in the vent fluid itself. Everything else living at the vent, the shrimp, the crabs, the worms, the fish that pass through, depends ultimately on this microbial base. The ecosystem is energetically sealed off from the sun. It runs on the chemistry of the earth. This is what makes hydrothermal vents one of the most philosophically significant discoveries in the history of biology. Before vents were first discovered, in 1977 at the Galapagos Rift, the assumption was that all life on Earth ultimately depended on sunlight. Photosynthesis was the foundation, and everything else was downstream of it. The discovery of chemosynthetic ecosystems at vents shattered that assumption. Life, it turned out, did not need the sun. It needed chemistry and energy, and the Earth's interior could supply both. The implications of that discovery have only grown more significant in the decades since. At the Cayman Trough's vent fields, the chemosynthetic communities are both familiar in their basic structure and genuinely novel in their specific inhabitants. The 2010 expedition documented several species that had not been seen before, and some of them were startling. The most immediately striking were the shrimp, found swarming in enormous numbers over the surfaces of the BB chimneys, sometimes in aggregations so dense that they formed a living crust on the vent structures. These shrimp were identified as a new species and formally described in 2012 as Remicarus hybisae. The name acknowledges both their location and the vehicle that found them, the ROV Hybis, which was used during part of the expedition. Remicarus hybisae is related to another vent shrimp, Remicarus exoculata, which has been well studied at mid-Atlantic ridge vent systems. But there are differences. Remicarus exoculata lives at depths of around 2,000 to 3,500 meters at Atlantic vents. Remicarus hybisae was found living at nearly 5,000 meters under significantly higher pressure. Like its relative, it appears to have modified eyes, not conventional image-forming eyes, but photoreceptive organs that may be sensitive to the faint glow of thermal radiation emitted by the superheated vent fluid. In the absolute darkness of the deep sea, even that faint non-solar light source may be useful for navigation. The shrimp feed on the chemosynthetic bacteria growing on the chimneys, scraping microbial mats from the mineral surfaces with specialized mouthparts. They live their entire lives in the zone of influence of the vents, and they exist in numbers that suggest the BB field is a productive and stable environment, at least on the timescales relevant to a shrimp. At the Von Dam field, the biological community looked different. The lower temperature, methane-rich diffuse flow at Von Dam supports a different set of organisms. Among the most visually distinctive were the crabs. Scientists found a species of kiwa, the genus that includes the so-called yeti crab, a group of hairy-chested crustaceans first described from Pacific vents in 2005. The Cayman species, informally nicknamed the Hoff crab after a somewhat affectionate comparison to a certain television actor known for a notably hairy chest, was found in dense aggregations on the flanks of the Von Dam field. Like other kiwa species, it appears to cultivate chemosynthetic bacteria on the dense bristles covering its body, effectively farming its own food source. The bacteria grow on the setae, the hair-like structures on the crab's claws and body, and the crab may feed on them directly, or benefit from their metabolic activity in other ways. The precise relationship is still being studied, but the basic picture is of an animal that has turned its own exterior into a microbial garden. These are not obscure, barely visible organisms. The shrimp swarms at BB and the crab aggregations at Von Dam are visible, numerous, and biologically significant. They represent the top of a food chain built entirely on chemical energy, and they were found on the very first dedicated scientific visit to these vent fields, which raises the obvious question of what else was there that the expedition did not find, or could not collect, or simply did not have time to document. The 2010 expedition was not a leisurely survey. Ship time in the deep ocean is expensive, and the RRS James Cook had a finite window to work in the Cayman Trough. The ROVs had limited bottom time at these depths, and the priority was to confirm the existence of the vents, collect samples, and gather as much data as possible within the available time. The biological collections from that expedition were remarkable given the constraints, but they were also incomplete by necessity. Certain areas of the vent fields were photographed but not sampled. Certain organisms were observed but not collected. The sediment around the vents, which in other vent systems has been found to harbour distinct microbial communities and small invertebrates, was not systematically sampled. The walls of the chimneys themselves, which can host layered microbial communities at different temperature gradients, were only partially examined. And then there is the question of what exists between the two vent fields and below them. The BB field sits at roughly 4,960 metres. The deepest point of the Cayman Trough is at approximately 7,686 metres. That is a difference of more than 2,700 metres of unexplored water column and seafloor. In that space, between the deepest known vent field and the actual bottom of the trough, there may be additional vent activity, or there may not be. There may be biological communities living on the sediment plains of the deeper trough, scavenging organic material that drifts down from above, or there may be very little. No one knows because no one has looked. That is not a rhetorical flourish. It is a straightforward statement of the current state of knowledge. The biological survey of the Cayman Trough below 5,000 metres is, for practical purposes, non-existent. The Hadal zone of the trough, the region below 6,000 metres where pressure and cold combine in their most extreme form, has never had a biological lander deployed in it, has never been visited by a crewed vehicle, and has never been the target of a dedicated scientific expedition. Whatever lives there, if anything does, has never been seen by a human being or a human-made instrument. There is a tendency, when confronted with facts like this, to assume that the absence of exploration implies the absence of life. If nothing has been found, perhaps there is nothing to find. But the history of deep-sea biology argues strongly against this assumption. Almost every time scientists have visited a new deep environment with the right equipment, they have found life, sometimes surprising life, sometimes life that required new categories to describe, the deep ocean has consistently exceeded expectations, and the Cayman Trough, with its active geology and its chemically rich vent systems, is not a place that gives obvious reasons to expect otherwise. The two vent fields already documented, Beebe and Von Dam, were found on the first serious attempt to look for them. The creatures living at those vents were new to science. The deeper sections of the trough have not been examined at all. The logic of that sequence is not complicated, and it points in one direction. There is almost certainly more to find. The question is not really whether undescribed species exist in the deeper Cayman Trough. The question is what they look like, how they live, and what they can tell us about the range of biological possibility on this planet. That question, quiet and patient as it is, has been sitting at the bottom of the Caribbean for a very long time, and the creatures living down there, whatever they are, have been going about their lives entirely without reference to whether we know about them or not. The events keep flowing, the bacteria keep oxidizing sulfide, the shrimp keep swarming in the dark, and somewhere deeper, in the parts of the trough that no instrument has reached, other organisms, shaped by the same pressures and the same chemistry, but perhaps in ways we have not yet imagined, are doing the same. What those organisms actually are, and what their existence would mean for our understanding of life in extreme environments, is where the story becomes genuinely remarkable. So let us talk about what has actually been found. Because before we can properly appreciate what remains unknown, it helps to sit with what the expeditions did manage to document. The creatures already confirmed in the Cayman Trough are remarkable enough on their own terms, and they deserve more than a passing mention. The most numerous animal discovered at the Beebe vent field was a shrimp, formerly described in 2012 as Rimicaris hybissae. If you have ever seen photographs of Atlantic hydrothermal vents, you may already be familiar with its close relative, Rimicaris exoculata, which swarms in dense masses on mid-Atlantic ridge vents. The Cayman species follows a similar pattern. At Beebe, the shrimp were found in aggregations of thousands, sometimes tens of thousands, covering the surfaces of the black smoker chimneys in a living, writhing layer. The density was described by researchers as almost carpet-like in places. Individual shrimp are small, a few centimetres long, pale, and in the way of many deep-sea crustaceans, essentially eyeless in the conventional sense. But Rimicaris hybissae, like its Atlantic cousin, appears to have modified photoreceptors on its back, structures that may be sensitive to the faint thermal glow emitted by superheated vent fluid. Whether this functions as a navigation tool, helping the shrimp stay close to the vents without touching the lethal hot water directly, is one of several open questions about its biology. What makes Rimicaris hybissae particularly interesting from a scientific standpoint is not just its presence at the deepest known vent system on Earth, but the degree to which it differs from its relatives. Genetic analysis confirmed it as a distinct species, and its adaptations to the extreme pressure at nearly 5,000 metres depth appear to go beyond what would be expected from a simple geographic variant. The pressure at Beebe is roughly 500 atmospheres, which is the kind of number that requires a moment to absorb, 500 times the pressure at sea level. At that pressure, the biochemistry of living cells faces real physical challenges. Proteins can denature, membranes can change their behaviour, enzymatic reactions that work perfectly well near the surface may function poorly or not at all. The organisms that thrive at these depths have evolved molecular adaptations, often at the level of individual protein structures, that compensate for these effects. In Rimicaris hybissae, those adaptations are almost certainly present, though the full picture of its deep pressure biochemistry has not yet been worked out in detail. The 2012 description paper established the species and noted its morphological differences from Atlantic relatives, but the molecular biology of its pressure tolerance remains an active area of interest. The shrimp were not alone at Beebe. Researchers also documented chemosynthetic bacteria forming thick mats on and around the vent structures, These bacteria are not incidental, they are the foundation. In a vent ecosystem, there is no sunlight, and therefore no photosynthesis, and therefore no plant life in the conventional sense. The entire food web rests on microorganisms that draw energy from chemical reactions, primarily the oxidation of hydrogen sulfide, methane, and other compounds that emerge from the seafloor with the vent fluid. The bacteria fix carbon using this chemical energy, producing organic matter that other organisms can then consume. It is a closed loop, energetically speaking, entirely independent of the sun. The bacteria at BB were found both as free-living mats on the seafloor and as symbionts living inside the tissues of larger animals, a pattern seen at vent systems around the world. Moving from BB to the Vondam vent field, which sits at a somewhat shallower depth of around 2,300 metres on the flanks of an underwater mountain called the Mount Dent, the biological picture shifts somewhat. Vondam is a different kind of vent system. Rather than producing the dramatic black smoker chimneys associated with the highest temperature vents, it generates a more diffuse, lower temperature flow that seeps through a broad area of carbonate and sulfide rock. The temperatures are still well above ambient, but the fluid chemistry and the physical structure of the field are distinct from BB. This difference in conditions produces a different community of organisms. The most visually striking creature documented at Vondam was the Kiwa species mentioned earlier, the hairy-chested crab that researchers informally called the Hoff crab. It belongs to the same genus as the famous Yeti crab, Kiwa hirsuta, first described from a Pacific vent in 2005. The Cayman Trough species has not yet been formally named as of the most recent published literature, which is itself a small indication of how much work remains to be done. The crab was found in aggregations on the Vondam field, and like other Kiwa species, it carries dense bristles called setae on its claws and the ventral surface of its body. These setae are colonized by chemosynthetic bacteria, and the crab appears to cultivate this microbial growth deliberately, positioning itself in areas of diffuse vent flow where the bacteria can thrive, and then apparently harvesting them. Whether it feeds directly on the bacteria by scraping them with specialized mouth parts, or benefits from them in some other metabolic way, is still not entirely resolved. The image of a small pale crab tending its own bacterial garden in total darkness at the bottom of the Caribbean is one of those facts that sounds like it should be from a science fiction novel, but is simply what the ocean does when left to its own devices for long enough. Researchers also noted the presence of other invertebrates at Vondam, including polychaeta worms, which are a common feature of vent ecosystems globally, and various gastropod mollusks. Snails and limpets adapted to vent conditions have been found at numerous deep-sea vent sites around the world, and their presence at Vondam follows an established pattern, though the specific species present in the Cayman Trough may well be distinct from those found elsewhere. The biogeographic isolation of the trough from other Atlantic vent systems means that even creatures belonging to familiar groups may have diverged enough to represent new species. In deep-sea biology, the rule of thumb is that if you visit a new vent field, you will find new species. The Cayman Trough has not been an exception to that rule. There were also observations of larger animals in the water column above the vents and in the surrounding deep water. Fish were recorded during the 2010 expedition, though systematic fish surveys were not the primary focus of the dive program. Some of these observations were of animals that could not be identified to species from video footage alone, which is a common frustration in deep-sea work. A fish seen briefly on camera, in poor visibility, at extreme depth, may be a known species, or it may be something entirely new. Without a physical specimen, it is often impossible to say. The ROV cameras used in deep-sea work have improved enormously over the past 2 decades, but the combination of low light, particle-filled water, and the tendency of animals to move away from the vehicle still makes definitive identification difficult in many cases. What the 2010 expedition established, taken as a whole, was a baseline, a first sketch of what the Cayman Trough's vent ecosystems look like. Shrimp in enormous numbers at the deepest vent field ever found. crab species cultivating bacterial gardens at the shallower field, chemosynthetic bacteria forming the energetic foundation of both communities, worms, mollusks and unidentified fish in the surrounding water, a food web operating entirely without sunlight, sustained by the heat and chemistry of the Earth's interior. That baseline is genuinely remarkable, and it was assembled in a very short time, on a very tight schedule, during the first visit anyone had ever made to these specific locations. But a baseline is also, by definition, incomplete. It is the beginning of a description, not the end of one. And what the 2010 expedition could not do, given its time constraints and the sheer size of the trough, was anything approaching a comprehensive survey. The 2 vent fields were the primary targets, and even within those fields the coverage was partial. The areas between the fields, the deeper sediment plains, the walls of the Rift Valley and the entire Hadal zone below 6,000 metres were not sampled. Some of these areas were not even imaged. It is worth being concrete about the scale of what was not visited. The Cayman Trough stretches roughly 1,400 kilometres in length. The 2 vent fields that have been studied are relatively small features within that larger geography. The BB field covers an area that can be measured in tens of metres across the most active zones. The Von Dam field is somewhat more spread out, but still occupies a limited footprint on the seafloor. The rest of the trough, the vast majority of its area, has been seen only in sonar maps, which tell you about the shape of the seafloor but nothing about what lives on it. Sonar mapping is genuinely useful. It can identify features like vent chimneys, lava flows, and sediment accumulations that suggest biological activity, but it cannot see organisms. It cannot distinguish a seafloor covered in dense microbial mats from a bare rock surface. It cannot detect a fish swimming 2 metres above the bottom. The gap between a sonar map and a biological survey is enormous, and in the Cayman Trough, that gap has barely begun to be closed. The 2010 expedition found new species on its first visit. The 2013 follow-up expedition found additional biological complexity at the same sites. Each visit has added to the picture without completing it. The sections of the trough that have not been visited at all, which is most of it, remain genuinely unknown in the biological sense. Not poorly understood, not partially documented. Unknown. This matters because the Cayman Trough is not a uniform environment. The geological processes that created it, the spreading ridge, the transformed faults, the deep rift valley, produce a landscape with significant variation. There may be additional vent fields along the spreading centre that have not yet been located. There may be cold seeps, places where methane and other compounds leak slowly from the sediment without the heat of a hydrothermal vent, which can also support chemosynthetic communities. The sediment plains of the deeper trough may harbour communities of small invertebrates and microorganisms adapted to the Hadal conditions there. None of this has been investigated. The creatures already documented at BB and Von Dam were found because scientists went looking for them, with the right equipment, in the right places. They were found quickly, on the first visit, because the vent fields are biologically rich, and the organisms living there are numerous enough to be immediately visible. The deeper and more diffuse habitats of the trough, the sediment plains, the rift walls, the Hadal floor, would require different tools and different strategies to survey. Biological landers, which are weighted frames dropped to the seafloor to collect scavenging organisms attracted by bait, have been used effectively in other deep trenches to document Hadal fish and crustaceans. None have been deployed in the Cayman Trough's deepest zones. The results of such deployments in other trenches have consistently produced surprises, and there is no particular reason to think the Cayman Trough would be different. The organisms already known from the trough are not the end of the story. They are, in a real sense, the introduction. They tell us that the trough is biologically active, that its vent systems support complex communities, and that those communities contain species found nowhere else on Earth. They also tell us, implicitly, that the sections of the trough that have not been examined are not likely to be empty. The logic of deep-sea biology, built up over decades of expeditions to other trenches and other vent systems, points consistently toward the same conclusion. Where there is energy, there is life. Where there is isolation, there is novelty. Where there is geology, this active, operating at these depths. For this long, the biological results are rarely simple or predictable. Which brings us to the physical environment itself, and why exploring the deeper sections of the trough is so much more difficult than visiting the vent fields that have already been studied. Because the conditions below 5,000 meters in the Cayman Trough are not merely extreme. They are extreme in ways that specifically challenge the equipment and methods that have worked at shallower depths. And understanding those conditions is necessary for understanding why the deeper trough has remained unvisited and what it would actually take to change that. Start with pressure. That is the most honest place to begin, because pressure is the first thing that changes when you descend below the surface of any ocean, and in the Cayman Trough, it changes in ways that are difficult to fully picture from where most of us are sitting right now. At sea level, the atmosphere pushes down on everything at roughly 1 atmosphere of pressure. That is the baseline, the number against which everything else is measured. Every 10 meters of seawater you descend adds approximately 1 more atmosphere. So at 100 meters depth, you're dealing with 11 atmospheres. At 1,000 meters, roughly 100 atmospheres. By the time you reach the deepest point of the Cayman Trough, at approximately 7,686 meters, the pressure has climbed to somewhere around 770 atmospheres. That is roughly 11,300 pounds per square inch, pressing inward from every direction simultaneously. To put that in terms that are easier to feel, it is approximately the equivalent of having 50 jumbo jets stacked on top of a single square meter of surface. The number is almost too large to be useful, but it is real, and it governs everything that happens at that depth. What 770 atmospheres does to equipment is not subtle. Standard research instruments are typically rated to around 600 atmospheres, which corresponds to depths of roughly 6,000 meters. Below that threshold, the engineering requirements change significantly. Pressure housings must be thicker, heavier, and more precisely manufactured. Seals that work perfectly at 4,000 meters can fail at 7,000 meters in ways that are catastrophic and irreversible. Electronics behave differently under extreme compression. Certain materials that are flexible at normal pressure become rigid or brittle at depth. Others that are rigid can crack. Syntactic foam, the material used to provide buoyancy to deep-sea vehicles, must be specially formulated for Hydrothermal depths, because standard foam compresses under pressure and loses its buoyancy, which is exactly the opposite of what you need when you are trying to bring something back to the surface. The engineering challenge is not just about making things strong enough to survive the pressure, it is about making them strong enough while keeping them light enough to be deployable from a ship, maneuverable at depth, and capable of carrying useful instruments. Every increase in pressure rating tends to mean more mass, which means more cost, which means fewer vehicles exist that can actually do the job. Most of the world's research are OVs, the remotely operated vehicles that do the actual work of deep-sea exploration, are rated to 6,000 meters. There are only a handful of vehicles in the world capable of operating at full ocean depth, meaning the deepest points of any trench on Earth, and they are in high demand, expensive to operate, and not always available for a Caribbean expedition that might not be at the top of the global priority list. The ROV Isis, which was used during the 2010 and 2013 Cayman Trough expeditions, has a rated depth of around 6,500 meters. That is deep enough to reach the BB vent field at approximately 4,960 meters and deep enough to conduct surveys in much of the trough. But the deepest point of the trough sits more than a kilometre below ISIS's operational limit. The Hydrothermal zone of the Cayman Trough, the portion below 6,000 meters, is outside the reach of the vehicles that have actually been used there. This is not a minor logistical detail. It means that the deepest floor of the trough has never been reached by any research vehicle, and cannot be reached without deploying equipment that is far rarer and more expensive than what has been used so far. Beyond pressure, there is the question of light, or more precisely, the complete absence of it. Sunlight penetrates the ocean to a maximum depth of around 200 meters under ideal conditions, and meaningful photosynthesis becomes impossible below about 100 meters. Below 1,000 meters, the ocean is in total permanent darkness. Not dim, not twilight. Absolute darkness. The kind that no amount of waiting allows your eyes to adjust to, because there is genuinely nothing there for them to work with. The Cayman Trough's floor sits nearly 8 times deeper than the point where darkness begins. Any light present at those depths is either bioluminescent, produced by organisms themselves, or artificial, brought down by human equipment. This matters for exploration in several ways. ROVs and landers must carry their own lighting systems, which consume power and add weight. The cameras used in deep-sea work must be sensitive enough to capture useful images in the narrow cone of light that the vehicle's own lights produce, while the rest of the environment remains invisible. Wide-area surveys of the kind you might do with aerial photography are simply not possible. You can only see what your lights illuminate, which means exploration at depth is always intimate and local in a way that surface exploration is not. You're never looking at the big picture. You're always looking at a small lit circle in the middle of an enormous darkness, moving slowly across a seafloor you cannot see until you are almost on top of it. The implications for biological survey work are significant. If an organism is not in the path of the vehicle's lights, it will not be detected. If it moves away from the vehicle before the cameras can capture it, it will not be recorded. If it lives in a crevice, under a rock, or buried in sediment, it may be completely invisible to a passing ROV. Deep-sea biologists are aware of this limitation, and they design surveys to account for it as much as possible, but there is no way to fully compensate for the fact that you are searching a vast, dark environment with a small moving flashlight. Temperature adds another layer to the picture. Away from the hydrothermal vents, the water near the floor of the Cayman Trough is cold. Not dramatically cold in the way that Arctic surface water is cold, but cold in the stable, relentless way of the deep ocean, somewhere between 2 and 4 degrees Celsius. This near freezing temperature is consistent across most of the deep ocean floor globally, because the cold, dense water that sinks near the poles spreads slowly across the ocean basins, filling the deep with water that has not seen sunlight in decades or centuries. The Cayman Trough is filled with this water in its ambient zones, far from the heat of the vents. The thermal contrast between the vent fields and the surrounding trench floor is therefore enormous. At the BB vents, temperatures exceed 400 degrees Celsius at the vent exit. A few hundred meters away, the ambient water is barely above freezing. Organisms living at the edges of vent influence must be adapted to navigate that gradient, and the communities that exist in the transition zones between hot vent fluid and cold ambient water are among the least understood in the entire system. Sound propagation at depth is also different from what we experience at the surface, though in ways that are more relevant to sonar mapping than to biological survey. Sound travels faster and farther in cold, pressurized water than in warm surface water, which is one reason that sonar is an effective tool for mapping the deep sea floor. But the acoustic environment of the Cayman Trough is also shaped by the geological activity of the spreading ridge, which produces low-frequency seismic noise that can interfere with some acoustic instruments. The trough is not a quiet environment in the acoustic sense, even though no human ear has ever heard it. The narrow width of the trough, roughly 80 kilometers at its widest points, creates additional navigational challenges for deep-sea vehicles. The walls of the Rift Valley are steep, in some places nearly vertical, and they are geologically active. The Mid-Cayman Spreading Centre is still moving, still producing new seafloor, still generating the occasional seismic event. The seafloor itself is not uniform. It is covered in a mix of basaltic rock, hydrothermal mineral deposits, and soft sediment, and the terrain varies considerably across short distances. Near the spreading ridge, the seafloor can be extremely rough, with lava formations, collapsed lava tubes, and fresh volcanic rock that has not yet accumulated any sediment cover. In the deeper rift axis, sediment accumulates in pockets between rocky outcrops, creating a complex mosaic of different substrate types. For a remotely operated vehicle, this terrain is genuinely difficult to navigate. ROVs are not agile in the way that a fish or a submarine with a human pilot might be. They are tethered to a ship at the surface by a cable that is kilometers long, and that cable creates drag, limits maneuverability, and can snag on geological features. The pilot controlling the ROV from the surface is working with a significant time delay between issuing a command and seeing the result, because the video feed and control signals must travel the full length of the cable in both directions. In complex terrain, this delay can make precise navigation genuinely dangerous. An ROV that moves too close to a wall or gets its tether caught on a rock formation can be lost. And losing an ROV at 5,000 meters depth is not like losing a drone in a field. It is a multi-million-dollar piece of equipment that is simply gone. The 2010 expedition used the ROV ISIS along with the autonomous underwater vehicle AutoSub 6000 for some survey work. Autonomous vehicles like AutoSub are not tethered, which gives them more freedom of movement in complex terrain, but they also cannot be controlled in real time. They follow a pre-programmed mission, and if something unexpected happens, the vehicle must make its own decisions, which may or may not be the right ones. AutoSub 6000 conducted sonar mapping and some sensor measurements during the expedition, contributing to the overall picture of the trough's geology. But autonomous vehicles of that generation were not capable of biological sampling, or the kind of close-up imaging that reveals new species. They could tell you what shape the seafloor was, they could not tell you what was living on it. Ship time is perhaps the most constraining factor of all, because it is the one that translates every other challenge into a financial and logistical reality. Operating a research vessel in the deep Caribbean is expensive. A single day of ship time, including the cost of the vessel, its crew, its fuel, and the associated support infrastructure, can run anywhere from $50,000 to over $100,000 depending on the vessel and the nature of the expedition. A deployment of an ROV to 5,000 meters depth takes several hours just to reach the bottom and several more hours to return to the surface. That means a single dive might consume the better part of a day, with only a few hours of actual bottom time in between. Over the course of a 3-week expedition, the number of dives that can be completed is limited, and each one must be carefully planned to maximize scientific return. The 2010 expedition had specific targets, the vent fields that sonar mapping had identified as likely locations of hydrothermal activity. The scientists aboard were not able to simply cruise around the trough looking for interesting things. They had to make decisions about where to send the ROV based on prior data and scientific hypotheses, and they had to live with the fact that most of the trough would remain unvisited during their time there. This is not a criticism of the expedition. It is simply the reality of deep-sea science. Every expedition is a negotiation between what you want to know, what you have time for, and what your equipment can actually do. The result of all these constraints, the pressure limits of available vehicles, the total darkness, the complex terrain, the tethered navigation challenges, the cost of ship time, is that the Cayman Trough's deeper zones have remained genuinely inaccessible. Not inaccessible in the sense of being theoretically unreachable, but inaccessible in the practical sense that the resources required to reach them have not yet been committed. The Hadal floor of the trough, the deepest kilometre or so of the Rift Valley, has never been visited by any scientific vehicle. The sediment plains at intermediate depths between the vent fields have not been sampled for biology. The walls of the rift have been imaged only in fragments. What makes this situation particularly interesting is that the physical environment of the deeper trough is not just a barrier to exploration. It is also, in a very real sense, the reason that whatever lives there has been left undisturbed for so long. The same pressure that destroys standard research equipment is the pressure that has shaped every organism in those depths into something specifically adapted to survive it. The same darkness that makes survey work so difficult is the darkness that has been the permanent condition of life at those depths for millions of years. The same cold ambient temperature that makes the environment feel inhospitable by human standards is the temperature that organisms living there have never needed to adapt away from, because it has never changed. The physical conditions of the deep trough are not just extreme, they are stable. And stability over geological time is one of the things that allows biological communities to develop in complex and specific ways. The organisms that live in the hadal zones of other deep trenches, the snailfish, the amphipods, the microbial communities in the sediment, have all been found to be specifically adapted to conditions that would kill organisms from shallower water. Their proteins fold differently, their cell membranes have different compositions, their biochemistry has been tuned over many generations to function at pressures and temperatures that are lethal to almost everything else on Earth. There is no reason to think the Cayman Trough's deepest zones would be different. There is every reason, given what has already been found at the vent fields, to think that the biological communities in the unexplored sections of the trough are both real and genuinely novel. But the physical environment that makes them interesting is the same environment that makes reaching them so difficult. And that tension between the conditions that create biological novelty and the conditions that resist human exploration is at the heart of why the deeper trough remains unknown. Understanding the physical environment of the trough is also necessary for understanding the broader history of why it has been explored so rarely, and what that history of inattention actually means for what might still be waiting there. Because the Cayman Trough did not become one of the most understudied deep trenches in the world by accident. There is a history behind that absence, and it is worth tracing carefully. The history of scientific attention to the Cayman Trough is, in a quiet way, a history of assumptions. Assumptions about where the interesting things were, about which oceans deserved the most scrutiny, about what the deep Caribbean could possibly offer that the deep Pacific had not already shown more dramatically and at greater expense. Those assumptions shaped decades of research priorities, and the result is a trench that has spent most of modern oceanographic history in the background, noted on maps, measured in broad strokes, and then largely left alone. The earliest serious attempts to understand the depth of the Caribbean basin came in the mid-20th century, as sonar technology developed enough to give researchers a rough picture of what lay beneath the surface. Expeditions in the 1950s and 1960s produced early bathymetric charts that established the general shape of the Cayman Trough, confirmed its exceptional depth, and identified the presence of a spreading ridge structure within it. These were significant findings, but they were also the kind of findings that opened questions rather than answered them, and for a long time, the scientific community did not rush to follow up. Part of the reason was practical. The Caribbean is a complicated region to work in. It is surrounded by multiple nations, many of them with overlapping and sometimes contested maritime jurisdictions. Organising a research expedition that might cross into different exclusive economic zones, that might need permits from several governments, that might require diplomatic coordination alongside the purely scientific planning, adds a layer of logistical difficulty that some research teams simply chose to avoid by going elsewhere. The Pacific, for all its vastness, has clearer research corridors in certain areas. The deep Atlantic has its own well-established research programs. The Caribbean sat in between, geopolitically complex and for a long time perceived as scientifically secondary. There was also a broader assumption at work, one that shaped deep-sea research funding for decades. The Pacific trenches, particularly the Mariana Trench, had captured the scientific imagination early and held it firmly. The Mariana Trench reaches 10,935 metres at the Challenger Deep, making it the deepest known point on Earth, and it had the distinction of being visited by a crewed vessel as early as 1960. when Jacques Picard and Don Walsh descended in the bathyscaphe Trieste to the bottom of the Challenger Deep. That dive was a landmark moment in the history of exploration, and it anchored Pacific Trench research as the prestige frontier of deep-sea science for generations. By comparison, the Cayman Trough, at roughly 7,686 metres, was deep but not the deepest. It was geologically interesting but not as immediately famous. It had no equivalent of the Trieste dive to give it a founding moment in the public imagination. It was, in the language of research funding, harder to justify as a priority when there were already well-established programs focused on more famous trenches. And so, decade after decade, the Trough received attention mostly in passing, as part of broader Caribbean geological surveys, or as a reference point in papers about Atlantic basin tectonics. Dedicated biological exploration of its depth was, for most of the 20th century, essentially non-existent. The first serious biological sampling of the Trough was minimal and largely incidental. Occasional trawl samples from intermediate depths captured some organisms, but these were not systematic surveys, and the deeper zones of the Trough were not targeted. The creatures that came up in those early nets were noted, catalogued if they were clearly identifiable, and filed away. There was no coordinated effort to understand what the deep Cayman Trough specifically contained, no program designed around the question of what lived there and why. It is worth pausing on that for a moment, because it is genuinely remarkable. By the time the first dedicated scientific dive to the BB vent field took place in 2010, human beings had already walked on the moon, had sequenced the human genome, had built the International Space Station, had sent spacecraft to the outer edges of the solar system. And yet the deepest vent system in the Caribbean, sitting in a sea crossed by thousands of ships every year, had never been visited. Not because it was unknown to exist, but because no one had organised the resources to go there. The 2010 expedition, led by the National Oceanography Centre and conducted aboard the RRS James Cook, was in this sense not just a scientific voyage, but a correction of a long oversight. The team used sonar data and water column measurements to identify the likely locations of hydrothermal activity before deploying the ROV ISIS to conduct the actual dives. When they descended to the BB vent field and found it active, rich with life, and sitting at a depth that made it the world's deepest known hydrothermal vent system, the reaction within the scientific community was genuine surprise mixed with a certain quiet recognition that the Trough had been underestimated for a long time. Compare this to the Mariana Trench, and the contrast is striking. The Mariana Trench has been the subject of scientific expeditions since the 1870s, when the HMS Challenger first measured exceptional depths in that region during its famous global oceanographic voyage. Since the Trieste dive in 1960, the Challenger Deep has been visited multiple times by crewed and uncrewed vehicles. James Cameron descended in a solo submersible in 2012. The Five Deeps expedition visited in 2019. Biological landers have been deployed to collect organisms from the Hadal zone. Sediment cores have been taken, water samples have been filtered for environmental DNA. The Mariana Trench is still not fully understood, but it has received sustained, serious and well-funded attention over more than 60 years. The Cayman Trough, in that same period, had its first dedicated biological dive in 2010. That is not a small difference. It is a 60-year gap in scientific attention between two deep trenches, one of which happens to be less famous than the other. The reasons for this gap are not mysterious, but they are worth naming clearly. Funding for deep-sea research follows attention, and attention follows prior discovery. The Mariana Trench had the Trieste dive, which generated enormous public interest and scientific momentum. That momentum attracted more funding, which produced more expeditions, which generated more discoveries, which attracted more funding still. The Cayman Trough had no equivalent catalyst. It sat in the Caribbean, deep and quiet and largely ignored, while the scientific world looked elsewhere. There is also the matter of perception about biological richness. For much of the 20th century, there was a working assumption among some researchers that the deep Atlantic was less biologically diverse than the deep Pacific, partly because the Atlantic is younger as an ocean basin, and partly because its deep zones were thought to be less geologically active and therefore less likely to support the kind of vent-based ecosystems that had been discovered in the Pacific in the late 1970s. The discovery of hydrothermal vents at the Galapagos Rift in 1977 had transformed the understanding of deep-sea life, but that discovery was in the Pacific, and for a time it reinforced the idea that the Atlantic Deep was a quieter, less eventful place. The Cayman Trough's vent fields have now challenged that assumption directly. The BB vent field is not a minor geological curiosity. It is the deepest known hydrothermal vent system on Earth, and it hosts a distinct biological community with species found nowhere else. The Von Dam vent field, at a shallower depth, adds a second distinct ecosystem within the same trench. Together, they suggest that the deep Caribbean is not biologically impoverished. It is biologically unstudied, which is a very different thing. What is perhaps most striking about the 2010 expedition is not just what it found, but what it implied about everything that had not yet been looked at. The BB vent field was identified through sonar anomalies and water column plume data before the ROV ever descended. It was not a lucky accident. It was a predicted target that turned out to be exactly as significant as the data suggested it might be. Which raises an obvious question. If the sonar data pointed to BB, what else does the sonar data point to in the sections of the trough that have not yet been followed up? Bathymetric surveys of the trough have identified other areas of geological interest, other sections of the spreading ridge, other potential sites of hydrothermal activity. Some of these have been noted in published papers. None of them have had an ROV descend to investigate. They sit on maps as interesting features, waiting for an expedition that has not yet been funded or organised. The scientific literature on the Cayman Trough is honest about this. Researchers who have worked there tend to write carefully about what has been sampled and what has not, and the lists of what has not been sampled are long. This is not unusual in deep-sea science. The ocean is large and research resources are finite, and every team working in every trench faces the same basic problem of having more questions than dives. But the Cayman Trough's particular history of neglect means that the gap between what is known and what might be there is larger than in more heavily studied systems. The Mariana Trench still has unknown biology, certainly, but it has been sampled enough that researchers have a reasonable sense of the broad outlines of its Hadal community. The Cayman Trough's Hadal zone has not been sampled at all. The outlines are completely unknown. There is something both humbling and quietly exciting about that. Humbling because it means that a significant portion of Earth's deep ocean remains genuinely terra incognita, even in the 21st century, even in a sea that humans have been sailing across for 500 years. And quietly exciting because it means that the discoveries still waiting in the trough are not incremental refinements of existing knowledge. They are likely to be genuinely new species, new communities, possibly new biological strategies for surviving conditions that most of life on Earth never encounters. The history of inattention to the Cayman Trough is, in this sense, a kind of preservation. The creatures living in its deeper zones have not been disturbed by repeated expeditions, have not been stressed by the noise and light of ROV visits, have not had their sediment disrupted by sampling equipment. They have simply been there, doing whatever they do, entirely unaware that the surface world has been largely unaware of them. Whether that is a good thing or a bad thing depends on what you think science should be for, but it does mean that when researchers eventually return with better equipment and more time, they will be arriving in a place that has been, in a meaningful sense, left intact. Understanding how rarely the trough has been visited, and why, also helps explain why the question of what lives in its unexplored zones is not a casual one. It is not a gap that will be filled by simply looking more carefully at existing data. It requires new expeditions, new dives, new equipment, and new funding. And it requires a shift in the perception that the deep Caribbean is somehow less worthy of that investment than the deep Pacific. The BB vent field alone should have been enough to make that case. Whether the broader scientific community and the funding bodies that support it will respond accordingly is a question that is still being worked out. What the history of Cayman Trough research makes clear, though, is that absence of evidence is not evidence of absence. The trough was not visited for decades, not because there was nothing there, but because no one went to look. When someone finally did go, they found a world record vent system teeming with previously unknown life. The logical implication of that finding is not that the rest of the trough has now been accounted for. It is that the rest of the trough might be equally surprising, and that the only way to know is to go deeper, stay longer, and look more carefully than anyone has managed to do so far. That question of what genuine biological novelty might be waiting in the trough's unexplored zones connects directly to something larger than the geography of a single Caribbean trench. It connects to a fundamental question about the nature of life itself, about how organisms adapt to conditions that seem designed to make survival impossible, and about what happens when a community of living things is left alone, in the dark, under extraordinary pressure, for a very long time. Because isolation and time in biology are not neutral forces. They are engines of change, and the Cayman Trough has had both in abundance. To understand why isolation and time matter so much in a place like the Cayman Trough, it helps to start with the basic mechanism that makes all of this possible. Because the creatures living around the BB and Von Dam vents are not surviving despite their environment. They are, in a very real sense, built by it. And the process that makes that possible is one of the more quietly remarkable things that biology has ever turned up. Chemosynthesis is the word for it, and it works like this. At the seafloor, superheated water exits the vent chimneys carrying dissolved compounds from deep inside the earth. Hydrogen sulfide is one of the primary ones. To most organisms, hydrogen sulfide is simply toxic. It is the compound that gives rotten eggs their smell, and in sufficient concentrations it will kill most life quickly. But certain bacteria have evolved the ability to do something different with it. Instead of being harmed by it, they oxidize it, stripping electrons from the sulfide molecules in a chemical reaction that releases energy. That energy is then used to fix carbon, to build organic compounds, to grow. It is, in its basic logic, not so different from what a plant does with sunlight. Capture an energy source, use it to build biological material, become food for something else. The difference is that there is no sunlight involved. There is no photon travelling 93 million miles from the sun. The energy comes entirely from the chemistry of the Earth itself, from the heat and the mineral-rich fluid bleeding out of the planet's interior. This matters enormously for what kind of life can exist in these places, because it means the food web at a hydrothermal vent has no connection whatsoever to the surface world. A shrimp swarming on a BB vent chimney is not even indirectly eating sunlight. It is eating bacteria that eat chemistry. The entire ecosystem is self-contained, powered by geology rather than astronomy. Which sounds abstract until you consider what it actually implies, that life in the deep ocean, around these vents, is not dependent on the sun being there at all. It would keep going in total darkness indefinitely, as long as the vents kept flowing. The BB vent field, sitting at nearly 5,000 meters depth, has been flowing for an unknown but geologically significant period of time. The Mid-Cayman Spreading Centre, the ridge system that drives the hydrothermal activity, has been spreading for millions of years. The vents themselves may not have been continuously active in exactly their current form for that entire span, but hydrothermal systems along active spreading ridges tend to be long-lived features, measured in thousands to tens of thousands of years at minimum, sometimes longer. That is a very long time for a community of organisms to develop, adapt, and become specialized. Now here is where biogeography comes in, and this is where things get genuinely interesting. The Cayman Trough's vent fields are not close to other hydrothermal vent systems. The nearest known active vent communities in the Atlantic are the Lost City Field near the Mid-Atlantic Ridge, and the TAG and Snake Pit fields further along the same ridge system. These are thousands of kilometers away. Between them and the Cayman Trough lies cold, deep, open ocean with no hydrothermal activity to serve as stepping stones. For the larvae of vent-dependent species, crossing that distance is not straightforward. Many vent organisms produce larvae that can disperse in ocean currents, but how far those larvae can travel before they need to settle and find a vent to survive on is limited. The cold, food-poor water of the deep ocean between vent fields is not a hospitable transit zone. What this means, practically, is that the communities at Beebe and Von Dam have likely been evolving in substantial isolation from other Atlantic vent communities for a very long time. The shrimp Rimicaris hibissae, discovered at Beebe in 2012, is closely related to Rimicaris exoculata, a species found at mid-Atlantic ridge vents. But the genetic analysis of hibissae showed clear divergence from its Atlantic relatives, divergence consistent with a long period of separate evolution. The two populations are related, but they have been on different paths for long enough that the Cayman species is now distinct, its own thing, shaped by the specific conditions of the Cayman Trough rather than by the conditions further east along the ridge system. This is not unusual in evolutionary biology, but it is particularly striking when you think about the scale of what it implies. A population of shrimp, or their ancestors, colonised the Cayman vents at some point in the past, possibly carried there by deep ocean currents, possibly dispersed during a period when the Atlantic ridge system was more geologically connected to the Caribbean. Once they arrived, the deep cold water between them and their relatives became an effective barrier. Generations passed. Thousands of them. Tens of thousands. Possibly more. The shrimp at Beebe adapted to the specific temperature gradients of those vents, to the specific chemical composition of the fluid, to the specific pressure and darkness of a depth that their mid-Atlantic cousins do not experience. And over time, they became something slightly different. Not unrecognisable, but distinct. Rimicaris hibissae is a species we know about because an ROV descended to Beebe in 2010, and its cameras captured images of thousands of them covering the vent chimneys in dense, writhing masses. But the Cayman Trough is large, and the sections of it that have been visited are small. If a population of shrimp could diverge significantly from its Atlantic relatives at a vent field that was only discovered 14 years ago, what might be happening at vent fields or geological features that have not been visited at all? This is not a rhetorical question. It is a scientific one, and it has a framework behind it. Biogeographic isolation combined with extreme environmental conditions is one of the most reliable drivers of biological novelty in the natural world. You see it on islands where populations cut off from mainland relatives evolve rapidly in response to local pressures, producing species found nowhere else. You see it in cave systems, where populations sealed off from surface ecosystems over thousands of years develop dramatic adaptations, losing pigmentation, losing eyes, developing extraordinary sensory systems to compensate. And you see it at hydrothermal vents, where the combination of extreme chemistry, extreme pressure, and extreme isolation creates communities that share basic evolutionary roots with the broader ocean but have been modified by their specific circumstances into something genuinely novel. The deeper zones of the Cayman Trough, below 5,000 metres, add another layer to this. Pressure increases significantly with depth. At the Beebe vent field, around 4,960 metres, pressure is already roughly 500 atmospheres. At the deepest point of the trough, 7,686 metres, it reaches approximately 770 atmospheres. That is not a small difference. The physiological challenges of surviving at Hadal depths, the deepest zone of the ocean, below 6,000 metres, are substantially greater than those at the depth of the Beebe vents. Proteins behave differently under extreme pressure. Cell membranes must be adapted to remain functional. Enzymes that work efficiently at moderate depths may lose their function, or change their structure at Hadal pressures. Organisms that live at these depths have evolved specific molecular adaptations to cope, including changes to the structure of their proteins and the composition of their cell membranes. What is interesting about the Cayman Trough is that it combines Hadal depth with the proximity of active hydrothermal systems. The BB vents sit at just under 5,000 metres, not quite Hadal depth, but close. The deepest zones of the trough, where no biological sampling has been conducted, sit well within Hadal range. If there are hydrothermal communities or chemosynthetically supported ecosystems at those depths, they would represent something that has not been documented anywhere, a vent ecosystem operating under full Hadal pressure. Whether such communities exist is genuinely unknown. The geological conditions and active spreading ridge within a deep trench suggest the possibility, but possibility and confirmation are different things, and the confirmation would require equipment and funding that have not yet been directed at the trough's deepest axis. Even setting aside the possibility of deep vent communities, the Hadal sediment plains of the trough are likely to harbour their own biological communities. In other Hadal zones, sediment communities are surprisingly rich. Amphipods, small crustaceans, appear in enormous numbers in Hadal trenches, often acting as scavengers that consume organic material sinking from the surface. Microbial communities in the sediment can be dense and metabolically active, processing organic carbon through pathways that are still being characterised. Hadal snailfish, like Pseudoliparis sweri in the Mariana Trench, have been found thriving at depths of 8,000 metres and more. Their bodies adapted in specific ways to survive pressures that would crush most fish. The Cayman Trough's Hadal zone, which is comparable in depth to the zones where these organisms have been found in the Pacific, has had none of this systematic attention. Whether its Hadal community resembles what has been found in the Mariana, or whether it has developed differently due to the Caribbean's distinct biogeographic history and the presence of active geology nearby, is simply not known. What makes the Cayman Trough specifically compelling, rather than just generically unexplored, is the combination of factors it brings together: extreme depth, active hydrothermal geology, Caribbean biogeographic isolation, proximity to the boundary between Atlantic and Pacific evolutionary lineages, and a history of minimal scientific attention that means the gap between what is known and what might be there is unusually large. Each of these factors on its own would make a place biologically interesting. Together, they create conditions that many researchers who have worked in the trough describe as a likely hotspot for undescribed species, not as a vague hope, but as a reasoned expectation based on what similar conditions have produced elsewhere. The genetic divergence already documented in Rimiceris hybesii gives this expectation some concrete grounding. If a shrimp population shows clear evolutionary separation from its nearest relatives after a period of isolation in the Cayman Trough, then other organisms in the trough, organisms that have been there longer, or that are more physiologically constrained and therefore less able to disperse, may show even greater divergence. Some of them may have diverged so far that they represent not just a new species but a new genus, or something even further removed from their nearest described relatives. This is not an unusual outcome in isolated extreme environments. It is in fact fairly typical. The question is simply whether anyone has looked. There is also the matter of metabolic novelty. The bacteria that form the base of chemosynthetic food webs at vents are not all the same. Different vent fields, with different fluid chemistry, different temperatures, and different mineral compositions, support different microbial communities. The specific bacteria at BB, oxidizing hydrogen sulfide at temperatures and pressures that would be exceptional even by vent standards, may have metabolic strategies not yet fully characterized. Some extreme offal bacteria have been found to use energy sources or chemical pathways that were not previously known to support life. The deep ocean has repeatedly surprised microbiologists in this way. The Cayman Trough's microbial communities, which have been only partially sampled even at the known vent fields, may hold examples of this kind of metabolic novelty. And in the unvisited zones of the trough, the microbial communities are entirely unknown. This is where the concept of biological dark matter becomes relevant. In the same way that astronomers use the term dark matter for mass that influences the universe but cannot be directly observed, Some microbiologists use a similar concept for the vast majority of microbial life that is known to exist, based on environmental DNA evidence, but has never been cultured or fully characterized. In deep-sea sediments, including hadal sediments, the proportion of microbial life that falls into this category is very high. The organisms are there, their genetic signatures are detectable, but what they actually do, what metabolic pathways they use, what ecological roles they fill remains largely unknown. The Cayman Trough sediment, unsampled in its deeper reaches, likely contains communities of this kind. What they are doing down there, in the cold and the dark and the pressure, is a question that has not yet been seriously asked, let alone answered. Isolation, then, is the thread that runs through all of this. Not isolation as emptiness, but isolation as a condition that allows biological trajectories to diverge, to specialize, to become strange in the best sense of the word. The Cayman Trough has been isolated from systematic scientific attention for almost its entire existence as a feature of the Earth's crust. Its biological communities have been isolated from other vent systems by thousands of kilometers of cold, deep ocean. Its hadal zones have been isolated from the surface world by kilometers of water and pressure. And within all of that isolation, life has been doing what life does, which is adapt, persist, and in the process become something that reflects its specific circumstances rather than anyone's prior expectations. The creatures in the trough's unexplored zones are not hypothetical in the sense of being invented or imagined. They are hypothetical only in the sense that they have not yet been confirmed. The conditions that produce biological novelty are present. The isolation is documented. The geological activity is measured. The timescale is geological. What remains is the confirmation, and the confirmation requires going there. Which brings the story naturally to what we actually know about the zones that have never been reached, and what science genuinely expects to find when someone finally does. So let us talk about what is actually down there. Or rather, what is not known to be down there, which is a different kind of conversation, and in some ways a more honest one. Below approximately 5,000 meters in the Cayman Trough, systematic biological sampling has not been conducted. That is not a rough estimate or a cautious understatement. It is simply the current state of the record. The ROVs deployed during the 2010 expedition and the follow-up work in 2013 were focused on the vent fields, which sit at depths between roughly 2,000 and 5,000 meters. The deeper trench axis, the sediment plains along the floor of the rift, the steep geological walls that drop toward the maximum depth of 7,686 meters, these areas have not had a biological lander placed on them. No crude submersible has visited them. No baited camera trap has been lowered to the deepest point and left to record what arrives. The hadal zone of the Cayman Trough, which begins somewhere around 6,000 meters by the conventional definition, is biologically uncharacterized. That is the honest baseline from which everything else in this section follows. It is worth pausing on that for a moment, because it can be easy to read a sentence like that and let it slide past without fully registering what it means. We are talking about a stretch of ocean floor in the Caribbean Sea, a body of water that has been sailed, fished, studied and crossed by human beings for centuries. The Caribbean is not a remote or poorly known ocean. It sits between continents that are densely populated and heavily trafficked. Cruise ships pass over the Cayman Trough regularly. Cargo vessels cross the region every day. The island of Grand Cayman, which gives the trough its name, is a well-known financial centre and tourist destination. And yet, directly beneath all of that familiar activity, there is a hadal zone that no biological instrument has ever reached. The contrast is genuinely striking, even if the explanation for it is straightforward enough. Depth is not the same as distance. The trough is close in terms of surface geography. It is effectively unreachable in terms of the engineering required to study its floor. What scientists hypothesize about the hadal zone of the Cayman Trough is informed by what has been found in comparable environments elsewhere, but it remains hypothesis. The clearest comparison is the Mariana Trench in the Western Pacific, which has received far more scientific attention over a longer period. In the Mariana, Hadal research has documented a surprisingly active community of organisms. Snailfish of the species Pseudoleparis sweri have been filmed and collected at depths exceeding 8,000 meters, which makes them among the deepest living vertebrates ever recorded. Their bodies show specific adaptations to Hadal pressure, including the accumulation of trimethylamine oxide, a compound that stabilizes proteins against the deforming effects of extreme compression. Amphipods, small crustacean scavengers, are present in the Mariana Hadal zone in very large numbers and appear to play a significant role in processing organic material that sinks from the surface. Microbial communities in the sediment are metabolically active and taxonomically diverse, carrying out chemical transformations in the cold and the dark that contribute to deep ocean nutrient cycling. Polychaete worms, holothurians, and other invertebrates have been documented at various Hadal depths. Whether any of this translates directly to the Cayman Trough is unknown. The trough is geologically different from the Mariana. It is a pull-apart basin rather than a subduction trench, which means the seafloor geology and the sediment character may differ in ways that affect what organisms can live there. The Caribbean's biogeographic history, its separation from the Pacific roughly 3 million years ago, and its distinct evolutionary lineages means that the species pool available to colonize the Cayman Hadal zone may be different from what colonized the Mariana. And the proximity of active hydrothermal geology within the same trench system introduces a chemical and thermal complexity that most Hadal environments do not have. The Mariana Trench is cold and geologically quiet at its floor. The Cayman Trough has a spreading ridge running through it and vent fields that alter the local chemistry of the water column. Whether that geological activity influences the Hadal community in the deeper parts of the trough, through hydrothermal plumes, through chemical gradients, through heat flow in the sediment, is a question that cannot be answered without going there. What researchers do expect, based on general principles of Hadal ecology, is that scavenging amphipods are probably present. They are found in essentially every Hadal environment that has been sampled with baited traps, and there is no obvious reason why the Cayman Trough would be an exception. Their presence would suggest that organic material from the surface is reaching the trench floor, which is likely given the productivity of Caribbean surface waters above the trough. If amphipods are there, then organisms that prey on them or compete with them for food may also be present. The food web in Hadal zones is not rich by shallow water standards, but it is not empty either. Hadal fish are another reasonable expectation. The snailfish family, Liparidae, has proven remarkably successful at colonizing Hadal depths across multiple ocean basins. Species have been documented in the Mariana, the Kermadec Trench near New Zealand, the Japan Trench, and several others. Their physiology appears well suited to Hadal conditions, and they have been found at depths that were previously considered incompatible with vertebrate life. Whether a snailfish species inhabits the Cayman Trough's deepest zones is unknown, but the family's broad Hadal distribution makes the possibility plausible. If one does exist there, it may be a species not yet described, given the trough's isolation from Pacific Hadal systems and its distinct Caribbean biogeographic context. Microbial life in the sediment is almost certain to be present. In every Hadal environment sampled so far, including some of the most extreme, sediment microbial communities have been found to be active and diverse. The sediment at the bottom of the Cayman Trough would accumulate organic material sinking from above and potentially receive chemical inputs from the hydrothermal system. Microbial communities capable of processing this material under high pressure and low temperature are well documented in other deep-sea sediments, and there is no reason to expect the Cayman Trough to be different in this fundamental respect. What is unknown is the specific composition of those communities, whether they include organisms with novel metabolic strategies, and how they interact with the unusual geochemistry of a trench that sits adjacent to an active spreading centre. The deeper walls of the trough are also essentially unstudied in biological terms. The steep rock faces that descend from the shallower flanks toward the trench floor may host sessile filter feeders, sponges, cold water corals, or other organisms that attach to hard substrate. These communities have been found on the walls of other deep trenches and on seamounts and ridges throughout the deep ocean. The Cayman Trough's walls, which in places drop thousands of meters over relatively short horizontal distances, provide a range of depths and substrate types that could support a variety of attached communities. None of this has been surveyed. The walls are steep enough and the terrain complex enough that ROV navigation along them would be challenging, but not impossible with the right equipment and sufficient ship time. It is also worth thinking about the transition zones, the areas between the known vent communities and the ambient deep sea environment. At the edges of vent influence, where hydrothermal fluid mixes with cold seawater and the chemical gradients become less extreme, communities of organisms have been observed that are distinct from both the vent fauna and the surrounding deep-sea fauna. These transition zones are ecologically interesting precisely because they represent a gradient of conditions from the chemically rich and thermally active vent environment to the cold, food-poor ambient deep sea. Organisms in these zones may be generalists capable of exploiting both environments, or specialists adapted to the specific conditions of the gradient itself. During the 2013 expedition, scientists observed animal communities at the edges of the Beebe and Von Dam vent fields that were not fully catalogued. Some were photographed but not collected. What they were, and how they fit into the broader ecology of the trough, remains an open question. Then there is the matter of the sediment plains between the vent fields. The floor of the Cayman Trough is not uniform. Between the areas of active hydrothermal geology, there are stretches of relatively flat sediment that receive no direct vent input. These areas are cold, dark and quiet. They may seem like the least interesting part of the trough, but deep-sea sediment plains have repeatedly surprised researchers who bothered to sample them. Soft-bodied invertebrates, meiofauna living between sediment grains, and microbial communities of considerable diversity have been found in sediment plains throughout the deep ocean. In the Cayman Trough, these intervent sediment areas have not been sampled at all. They sit between the vent fields, accessible in principle to a lander or a long ROV transect, but simply never visited. The absence of data from all of these zones is not itself evidence of anything in particular. It does not prove that remarkable creatures are hiding there, any more than an unread book proves it contains a masterpiece. What it means is that the question has not been answered. And in the deep ocean, where the conditions for biological novelty are well established, and where every serious sampling effort in a new environment has produced new species, the unanswered question carries genuine scientific weight. The Cayman Trough's deepest zones are not blank because they are empty. They are blank because no one has yet filled them in. There is a phrase that comes up in discussions of deep-sea biology with some regularity, and it is worth stating plainly here. The absence of evidence is not evidence of absence. It is a principle that applies with particular force to hadal environments, where the difficulty of sampling means that the absence of a record reflects the limits of the effort rather than the limits of the biology. In the Mariana Trench, the discovery of snailfish at 8,000 meters was not predicted with confidence before the observations were made. The fish were found because someone lowered a baited camera to a depth where many researchers assumed vertebrate life would be marginal at best. The camera recorded something unexpected. That is how deep-sea biology tends to work. You go, you look, and you find things that the prior expectation did not fully accommodate. The Cayman Trough's hadal zone has not had that moment yet. The baited camera has not been lowered to the deepest point. The lander has not sat on the trench floor for a day and recorded what arrives to feed. The sediment cores have not been pulled from the deepest sediment and analysed for microbial diversity. These are not technically impossible things. They are things that require a ship, the right equipment, funding, and the decision to prioritise this particular stretch of unexplored ocean over the many other places that also deserve attention. That decision has not yet been made in a sustained way. What makes this specific gap in the record feel significant rather than just one of many gaps in the ocean's enormous ledger, is the combination of factors that the trough brings together: the active geology, the documented vent communities with their already surprising inhabitants, the Caribbean biogeographic context, the isolation from other hadal systems, and the genetic evidence already in hand from Rimikare's Hybaceae. That isolation in this specific environment produces measurable evolutionary divergence. All of these things together create a scientific case for the Cayman Trough's deepest zones being not merely unstudied but specifically worth studying, for reasons that go beyond general deep-sea curiosity. The Hadal zone of the Cayman Trough may contain organisms that resemble what has been found in the Mariana. It may contain organisms that are genuinely different, shaped by the Caribbean's evolutionary history and the Trough's unusual geology. It almost certainly contains microbial communities that have not been characterized. It may contain fish, crustaceans, and invertebrates that have not been described, and it may contain things that do not fit neatly into any of these categories because the deep ocean has a way of producing forms of life that do not announce themselves in advance. None of this is certain. The honest position is that the Cayman Trough's Hadal zone is a place where the scientific expectation of biological novelty is well founded, but where the confirmation of anything specific requires going there and looking. And that, in turn, depends on what tools are available, what they can do, and whether the resources exist to deploy them in a place that is close in geography, but still very far away in terms of the effort required to reach its floor. Which is where the story moves next, into the question of what it actually takes to explore a place like this, and what the current state of deep-sea technology makes possible. So let us step back for a moment from the specific geography of the Cayman Trough and ask a broader question, one that gives the Trough's unexplored zones their proper context. How much of the deep ocean, as a whole, remains unknown? The answer is uncomfortable in a way that tends to surprise people when they first encounter it. Estimates vary depending on methodology and how you define a species as formally described, but some marine biologists working in deep-sea taxonomy suggest that fewer than 20% of deep-sea species have been catalogued. That is not a fringe position. It reflects the genuine state of the field, one where the rate of new discoveries has consistently outpaced the resources available to process and describe them. Taxonomists who specialize in deep-sea invertebrates often have backlogs of specimens collected years ago that have not yet been formally named, because naming a species properly requires detailed morphological work, genetic analysis, comparison with related species, and publication in a peer-reviewed journal. The ocean is producing new animals faster than the scientific infrastructure can formally absorb them. In 2023, the Ocean Census, a global initiative specifically designed to accelerate the discovery and description of marine species, identified over 800 new species in a single coordinated year of effort. 800 in 1 year. And that was a project that was not exclusively focused on the deep sea. It covered a range of marine environments. The number gives you a sense of the scale of what remains to be done, and it also gives you a sense of what becomes possible when resources and coordination are actually applied to the problem. The deep ocean itself covers more than 60% of Earth's surface. That is not a dramatic rhetorical flourish. It is simply the arithmetic of the planet. The ocean is large, and most of it is deep, and the deep portion is the least explored biome on Earth by a significant margin. We have better maps of the surface of Mars than we do of the ocean floor. That particular comparison has been made so many times that it risks feeling like a cliché, but it keeps being repeated because it keeps being true, and because it keeps being the kind of fact that takes a moment to actually settle in. The Cayman Trough exists within this broader context. It is not unique in being understudied. Almost all deep-sea environments are understudied. But what makes the Trough specifically interesting, rather than just another entry in the long list of places that deserve more attention, is the combination of factors that converge there and what those factors predict about the biology. The Caribbean sits at a biogeographic crossroads. This is worth explaining carefully because it matters for understanding what kinds of organisms might be living in the Trough's deeper zones. Roughly 3 million years ago, the Isthmus of Panama closed. Before that closure, there was a shallow seaway connecting the Atlantic and Pacific Oceans through what is now Central America. Marine species could move between the two ocean basins. After the isthmus closed, populations that had been connected were separated. They were cut off from each other by a land barrier that has not reopened in 3 million years. Over that time, species on either side of the divide diverged. The same ancestral population, split into two by geology, eventually became two distinct species, shaped by different conditions, different predators, different food sources, different temperatures. This process is called vicariance, and it has left a deep signature in the genetics of Caribbean marine life. For shallow water species, this signature has been studied fairly extensively. We can trace which Caribbean fish and invertebrates are most closely related to their Pacific counterparts, and we can use the genetic distance between them to estimate how long they have been separated. But for deep water species, particularly those living in trenches and vent systems, this kind of comparative work is in its early stages. The deep Caribbean has not been sampled thoroughly enough to know which deep-sea lineages carry the evolutionary signature of the Panama closure, or what other ancient separations might be written into their genomes. What some researchers believe, based on the general principles of biogeography and the limited data available, is that the Cayman Trough's deep zones may harbour species whose evolutionary history connects to that ancient Caribbean isolation. Deep water species often have slower dispersal rates than shallow water ones. They may be more dependent on specific substrate types, specific chemical conditions, specific temperature ranges. These constraints mean that deep-sea populations can remain isolated for very long periods, accumulating genetic differences that eventually produce new species. The trough's combination of depth, unusual geology, and Caribbean biogeographic position makes it, in the view of some researchers, a likely location for species with long evolutionary histories that have not yet been documented. This is not speculation in the pejorative sense. It is a prediction derived from a scientific framework, the same kind of prediction that led researchers to expect that the Galapagos Rift would harbour unusual life when hydrothermal vents were first proposed as a possibility there in the 1970s. The prediction was made on theoretical grounds before the vents were discovered. When the Alvin submersible visited the Galapagos Rift in 1977 and found tube worms, clams and dense biological communities around the vents, it confirmed a hypothesis that had been built on sound reasoning. The Cayman Trough is in a somewhat analogous position. The reasoning for expecting biological novelty there is sound. The confirmation requires the field work. There is also the matter of what the trough's existing vent fauna has already demonstrated about the potential for divergence in this specific environment. Remicarus hybissae, the shrimp found in enormous swarms at the BB vent field, is genetically distinct from its closest relatives at mid-Atlantic ridge vents. The genetic analysis published following its description in 2012 showed divergence consistent with a long period of isolation. The Cayman Trough's vents are separated from the nearest Atlantic vent systems by thousands of kilometres of cold, deep ocean. For a vent-dependent organism, that distance is effectively impassable. There is no stepping stone route between the Cayman vents and the mid-Atlantic ridge. Any population that colonised the Cayman vents in the distant past would have been cut off from the parent population, and natural selection would have gone to work on them in the specific conditions of the Beebe and Von Dam fields. The result, after enough time, is a shrimp that looks related to its Atlantic cousins but is not the same animal. It is its own species, shaped by its own place. If that kind of divergence has happened at the vents, which sit at roughly 5,000 metres depth, it is reasonable to ask what has been happening in the deeper zones of the trough below 6,000 metres, where conditions are even more extreme and where no sampling has been conducted at all. The Hadal zone, generally defined as the depth range below 6,000 metres, is a distinct environment from the bathyal and abyssal zones above it. The pressure is higher, the food supply from the surface is different, partly because of how organic matter sinks and accumulates in the narrow geometry of a trench. The fauna of Hadal zones tends to be distinct from the fauna of the surrounding abyssal plains, and Hadal faunas from different trenches are often distinct from each other, reflecting the isolation of individual trench systems from one another. Each Hadal zone is, in a sense, its own evolutionary island. The Mariana Trench, which is the most studied Hadal system on Earth, has yielded species that were not known before systematic Hadal sampling began in the mid-20th century. The snailfish Pseudoleperis suere, described formally in 2017 from specimens collected at depths between approximately 6,000 and 8,000 meters, is one example. Hadal amphipods, small crustaceans that scavenge on the trench floor, have been found in enormous numbers at bait stations in multiple Hadal zones, and different trench systems host different species assemblages. Microbial communities in Hadal sediments show high diversity and metabolic activity that researchers did not anticipate before detailed sampling was conducted. The lesson from the Mariana and from other well-studied Hadal systems is consistent. These environments are not biological deserts. They're inhabited, sometimes densely, by organisms specifically adapted to the conditions there. The Cayman Trough's Hadal zone, by contrast, has had none of this systematic attention. No baited camera has been lowered to the deepest point and left to record what arrives. No sediment cores have been taken from the trench floor and analysed for microbial diversity. No biological lander has sat on the bottom at 7,000 metres and waited. The deepest biological observations in the trough were made at the BB vent field, at approximately 5,000 metres during the 2010 and 2013 expeditions. Below that depth, in the trough's true Hadal zone, the biological record is essentially empty. That emptiness is not neutral. In the context of everything that deep-sea biology has produced in the last 50 years, an unsampled Hadal zone in a geologically active, biogeographically distinctive trench is a gap that carries scientific significance. It does not guarantee that remarkable things are there, but it means that the question is genuinely open in a way that is not true of environments that have been repeatedly sampled and found to be less diverse than expected. There is a particular quality to this kind of scientific uncertainty that is worth pausing on for a moment. In everyday life, uncertainty often feels like a problem, something to be resolved as quickly as possible so that decisions can be made. But in science, uncertainty of this kind, the uncertainty that comes from not yet having looked, rather than from having looked and found contradictory results, is actually a productive state. It is the state in which discovery is possible. The Cayman Trough's Hadal zone is uncertain in this productive sense. We do not know what is there. That means that when someone eventually goes and looks, they may find something. And in the deep ocean, the history of such moments is one of consistent surprise. Some researchers working in deep-sea biogeography have specifically identified the Cayman Trough as a priority site for future Hadal surveys, precisely because of the combination of factors described here. The active geology, the documented vent communities, the Caribbean biogeographic context, and the complete absence of Hadal biological data make it a place where the expected return on scientific investment is high. That does not mean the funding has materialised, or that the expeditions have been planned. Priority lists in science are long, and the resources available for deep-sea work are limited relative to the scale of the unexplored territory. But the scientific case for looking more carefully at the Cayman Trough's deeper zones is not a matter of wishful thinking. It is a reasoned argument based on what is already known. It is also worth noting gently that the Cayman Trough is not in some remote corner of the planet that requires extraordinary logistics to reach. It sits in the Western Caribbean Sea, in a region that is well served by ports in Jamaica, Cuba, the Cayman Islands, and nearby Central American countries. Research vessels operate regularly in Caribbean waters. The challenge is not access in the geographic sense. It is the combination of depth, which requires specialised equipment, and the cost of ship time and ROV operations at the depths required. These are real barriers, but they are not absolute ones. The Five Deeps expedition, which sent a crewed submersible to the deepest point in each of the world's 5 oceans between 2018 and 2019, demonstrated that Hadal exploration is operationally feasible when the resources and the will are present. The Atlantic Deep, specifically the Puerto Rico Trench, was one of the 5 Deeps targets. The Cayman Trough, deeper than the Puerto Rico Trench in its maximum depth, was not included. It remains, in that sense, a place that has not yet had its moment. What would that moment look like if it came? It would probably begin not with a crude submersible, but with a systematic survey using a combination of tools: an autonomous underwater vehicle mapping the trench floor in detail, a full ocean depth ROV or lander conducting biological observations and collections, and water column sampling for environmental DNA at multiple depths. The eDNA approach is particularly promising for a first survey because it can detect the presence of organisms without requiring physical capture. Filtering water at different depths and analysing the genetic material suspended in it can produce a preliminary inventory of what taxa are present, which then guides where to focus more intensive sampling. This technique has been used in other deep environments, including the Mariana Trench, and has consistently detected species that were not observed directly during the same dives. A systematic survey of this kind would not resolve every question about the Cayman Trough's biology. It would not instantly produce a complete species list or explain every ecological relationship in the trench, but it would transform the current situation, in which the Hadal zone is a complete blank, into one in which at least the broad outlines of what lives there are known. And in the deep ocean, that kind of first-pass knowledge tends to generate more questions than it answers, which is precisely what makes the field productive. The broader picture, then, is this. The Cayman Trough sits within a deep ocean that is, as a whole, far less known than the public perception of modern science might suggest. The tools to study it are improving. The scientific framework for predicting what kinds of novelty might be found there is well developed. The specific characteristics of the trough, its geology, its isolation, its biogeographic position, and its documented vent fauna all point toward it being a place where serious biological survey would produce meaningful results. And the Hadal zone of the trough, its deepest and least accessible portion, remains completely uncharacterized from a biological standpoint. That is the honest summary of where things stand. And that summary points naturally toward a different kind of question, one that is not just about what creatures might be hiding in the trough's depths, but about what it would actually take to find them. The technology exists in various forms and at various levels of development, but technology alone does not drive exploration. The history of deep-sea science is, in large part, a history of what happens when the right equipment meets the right funding, meets the right scientific moment. Understanding what those tools currently look like and what their limitations are is the next piece of this story. So the scientific case is made, the framework is in place, the tools are being developed, and yet, when you trace the actual history of expeditions to the Cayman Trough, what you find is a pattern that is both understandable and quietly frustrating. A handful of visits, each one producing remarkable results, each one leaving most of the trough untouched, and long gaps between them where the trench simply waited in the dark. The 2010 expedition was the first dedicated scientific dive to the BB Vent field. That was the breakthrough, the moment when the world's deepest hydrothermal vent system was formally visited and documented. But a single expedition, however successful, is not a survey. It is more like a first handshake. You learn that something is there. You learn a few of its features. You go home with specimens and data and a long list of new questions, and then the real work begins, which requires going back. The return came in 2013, aboard the same vessel that had made the original discovery. The RRS James Cook, a research ship operated by the Natural Environment Research Council in the United Kingdom, returned to the Cayman Trough on a cruise designated JC-08-2. The ROV Isis, a work-class remotely operated vehicle capable of operating at depths down to 6,500 metres, was once again the primary tool for reaching the seafloor. The scientific team, again led largely from the National Oceanography Centre in Southampton, had specific objectives. They wanted to extend the biological documentation of the Beebe and Von Dam vent fields, collect additional specimens for taxonomic and genetic analysis, and measure the chemical and thermal properties of the vent fluids in more detail. What they found when they returned to Beebe was in some ways reassuring, and in other ways quietly humbling. The vents were still active, the shrimp swarms were still there, dense aggregations of Rimicaris hibissae coating the surfaces of the vent chimneys in numbers that researchers estimated in the thousands per square metre. The chimneys themselves, those black smoker structures rising from the seafloor and venting superheated fluid, were structurally similar to what had been observed in 2010, though some had changed in subtle ways. Hydrothermal vent systems are not static. The flow rates shift, chimneys collapse and new ones form. The biological communities respond to these changes, redistributing themselves as the thermal gradients move. The team collected additional specimens of Rimicaris hibissae, which allowed for more detailed genetic and morphological analysis back in the laboratory. They also observed other organisms in the areas surrounding the main vent chimneys, in what researchers sometimes call the periphery or the transition zone, where the influence of hot vent fluid grades gradually into the cold, ambient water of the deep ocean. This transition zone is ecologically interesting precisely because it is not the extreme environment of the vent itself. The temperatures are lower, the chemical concentrations are more dilute, and the conditions are somewhat more tolerable for organisms that are not fully adapted to the vent environment. It is, in a sense, the border region between two very different worlds. In these peripheral areas, the 2013 expedition observed animal communities that had not been fully catalogued during the 2010 visit. Some of these organisms were filmed by the ROV's cameras. Some were photographed in detail, but not all of them were collected. This is a constraint that comes up repeatedly in deep-sea biology, and it is worth understanding clearly. An ROV like ISIS carries a manipulator arm and sample collection containers, but the number of specimens that can be physically retrieved during a single dive is limited. The arm can only reach so far, the containers can only hold so many samples, and the time on the bottom, which is already limited by the length of the umbilical cable and the endurance of the ship's dynamic positioning systems, does not allow for exhaustive collection across a wide area. So some of what was seen at the edges of the Beebe vent field in 2013 was documented visually but not physically captured. That means organisms were observed, their images recorded, but without a physical specimen, formal taxonomic description is not possible. You cannot name a new species from a photograph alone, at least not under the current rules of biological nomenclature. You can note that something unusual was present. You can estimate its size, describe its apparent morphology, note its behaviour and its position in the habitat. But the formal step of saying this is a new species. Here is its name, here is the type specimen deposited in a museum collection. That step requires tissue. It requires the animal itself, or at least a piece of it. This is not a minor procedural point. It means that there are organisms in the Cayman Trough that have been seen by human eyes, recorded on video, discussed in expedition reports, and still remain formally unnamed and undescribed. They exist in a kind of scientific limbo, observed but not yet known in the full sense. How many such organisms there are is difficult to say, but the 2013 expedition report suggests that the peripheral communities of the Beebe field alone contained more biological complexity than a single expedition could fully characterise. At Von Dam, the situation was somewhat different. This vent field, sitting at approximately 2,300 metres depth on the flanks of an underwater mountain called Mount Dent, is shallower and more accessible than Beebe. The lower temperature and more diffuse flow of Von Dam creates a different kind of habitat, one that supports different biological communities. The hairy-chested Kiwa crab, sometimes informally called the Hoff crab, after the famously chest-haired actor, had been found here during the 2010 expedition. In 2013, researchers were able to observe these animals more extensively and collect additional specimens. The Kiwa crabs at Von Dam appear to farm chemosynthetic bacteria on their bristled chest hairs, waving their claws through the water near the vents to cultivate the microbial growth that forms a significant part of their diet. It is a feeding strategy unlike anything seen in shallow water crustaceans, and it is a good example of how isolated vent environments can produce not just new species, but genuinely novel biological strategies. But even at Von Dam, the coverage was partial. The vent field extends over an area that is large relative to the footprint of a single ROV dive. The 2013 expedition conducted multiple dives, but each dive covered a specific section of the field. The areas between dives, the terrain that was not directly traversed by the ROV, remained unsampled. And beyond the vent fields themselves, the broader seafloor of the Cayman Trough, the sediment plains between the vents, the steep walls of the Rift Valley, the geological structures along the spreading centre, received essentially no biological attention during either the 2010 or 2013 expeditions. The expeditions were focused on the vents. Everything else was, in effect, background. This is not a criticism. It is simply how targeted scientific expeditions work. You go to the place most likely to produce results, you document it as thoroughly as your equipment and time allow, and you bring back what you can. The 2010 and 2013 expeditions to the Cayman Trough were scientifically productive by any reasonable measure. They discovered new species, established the existence of the world's deepest hydrothermal vent system, and produced a body of data that has informed subsequent research across multiple disciplines. The scientists involved did exactly what they set out to do. But the trough is large. It stretches for roughly 1,400 kilometres. The vent fields that were visited represent a small fraction of the total geological structure. The sediment plains, which in other deep-sea environments have been found to harbour their own biological communities, including microbial mats, small invertebrates, and scavenging fish, were not surveyed. The walls of the Rift Valley, which in other spreading centre environments can support diverse communities of sponges, corals, and associated fauna, were not explored. And the Hadal Zone, the deepest portion of the trough below 6,000 metres, was not reached at all by either expedition. The ROV Isis has a maximum operating depth of 6,500 metres, which means it could theoretically reach into the Hadal Zone of the Cayman Trough, but the expedition priorities kept it focused on the shallower vent fields. There is something worth sitting with in that fact. The deepest point of the Cayman Trough, at 7,686 metres, has never had a biological lander, a crude submersible, or an ROV conducting systematic observations anywhere near it. The Hadal Zone of one of the Caribbean's most geologically active and biologically interesting trenches is, from a biological standpoint, completely blank. Not poorly characterised, not sparsely sampled. Blank. In the years since 2013, there have been additional scientific publications based on the specimens and data collected during the two main expeditions. Genetic analyses of Rhimacharis hybaceae have been published, detailing how this shrimp differs from its relatives at other Atlantic vent sites, and what those differences suggest about the evolutionary history of the vent shrimp lineage. Geochemical studies of the BB vent fluids have been published, characterizing the unusual chemistry of the world's deepest black smokers. Microbial studies have examined the chemosynthetic bacteria at both vent fields. All of this represents genuine scientific progress, and it has been achieved through careful analysis of what was brought back from the 2010 and 2013 dives. But there have not, as of the most recent publicly available records, been further dedicated deep biological expeditions to the Cayman Trough of the same scale and depth capability as those two cruises. The scientific community is aware of the trough. The papers are cited, the species are known. But the follow-up survey that would extend coverage beyond the vent fields, and particularly into the Hadal Zone, has not materialised. This is partly a funding question, partly a question of competing priorities in a field where there are many unexplored places and limited ship time, and partly a question of equipment. A systematic survey of the Cayman Trough's deepest zones would require full ocean depth capability, which means either a lander system rated to 8,000 meters or a vehicle like the Limiting Factor, which was used in the 5 Deeps expedition and is one of the few crewed submersibles capable of reaching any depth in the ocean. The 5 Deeps expedition itself is instructive here. Between 2018 and 2019, explorer Victor Vescovo and a small scientific team used the Limiting Factor to reach the deepest point in each of the world's 5 oceans. The Atlantic Deep was represented by the Puerto Rico Trench, which reaches approximately 8,376 meters at its deepest point. The Cayman Trough, though it is also in the Atlantic Basin and reaches 7,686 meters, was not included in the expedition's targets. This is not surprising. The 5 Deeps was organised around 1 deep per ocean, and the Puerto Rico Trench is deeper. But it does illustrate the way in which the Cayman Trough tends to fall just outside the frame of the most ambitious exploration efforts. It is deep, but not the deepest. It is in the Atlantic, but not the Atlantic's deepest point. It is geologically remarkable, but the Mariana Trench is more famous. In a world where attention and resources are finite, being the second most notable example of something is often enough to be overlooked. And yet, what the 2010 and 2013 expeditions established is that the Cayman Trough is not simply a deep version of other deep places. The combination of active spreading geology, isolated vent ecosystems, Caribbean biogeographic position, and extreme depth makes it genuinely distinctive. The organisms found there are not just variants of what you would find in the Mariana Trench or the Mid-Atlantic Ridge. They are specifically Cayman Trough organisms shaped by the particular conditions of this particular place. Remicoris hybaceae is not found anywhere else. The kiwa crab population at Von Dam is genetically distinct from kiwa populations at other vent sites. The microbial communities at BB reflect the unusual chemistry of the world's deepest vent fluids. These are not generic deep-sea creatures. They are creatures of this trench, and the trench has more of them in its unvisited zones than any expedition has yet managed to find. That is the honest picture of where the exploration record stands. 2 main expeditions, remarkable results, partial coverage, and a Hadal zone that remains completely uncharacterized. The scientific community knows this. The researchers who have worked in the Cayman Trough know this better than anyone. And the knowledge that the most interesting parts of the trough, in terms of potential biological novelty, are precisely the parts that have not yet been reached, is something that sits quietly in the background of the published literature, present in the discussion sections of papers, in the recommendations for future work, in the grant applications that have been written and sometimes funded and sometimes not. What all of this points toward is a question that goes beyond the Cayman Trough itself. It is a question about the relationship between technology and exploration, about what the current state of deep-sea tools actually allows, and about what would need to change for the blank spaces in the trough's biological record to start being filled in. Because the barriers to deeper exploration of the Cayman Trough are not primarily scientific. The scientific case is clear. The barriers are technological and financial, and understanding them in detail is the next part of this story. So let us think about the technology more carefully, because this is where the gap between what we know and what we could know becomes very concrete. Most research ROVs currently in operation around the world are rated to a maximum depth of 6,000 meters. That number is not arbitrary. It represents a practical engineering threshold, the point at which the cost and complexity of building a vehicle to survive greater pressure begins to increase very steeply. At 6,000 meters, the pressure is already around 600 atmospheres, which is roughly 8,800 pounds per square inch. Every component on the vehicle, every camera housing, every thruster seal, every cable penetration, every electronic enclosure has to be engineered to survive that force without leaking, crushing, or failing. It is a significant engineering achievement just to get a vehicle to that depth reliably. Going deeper requires a different category of design entirely. The Cayman Trough's maximum depth of 7,686 metres puts its Hadal zone well beyond the reach of most of the world's operational research ROVs. The ROV ISIS, which conducted the 2010 and 2013 dives to the BB and Vondam vent fields, has a rated depth of 6,500 metres. That is enough to reach the vent fields, which sit at around 4,960 and 2,300 metres respectively, with considerable margin to spare. But the deepest axis of the trough, the Hadal floor where the Rift Valley reaches its maximum depth, is beyond ISIS entirely. You cannot simply push a 6,500 metre vehicle to 7,686 metres and hope for the best. The engineering does not bend that way. Full ocean depth vehicles, meaning submersibles and ROVs capable of reaching any point in the ocean, regardless of depth, exist, but they are rare. The Nereid Under Ice, developed by the Woods Hole Oceanographic Institution, is one example of a vehicle designed to operate at extreme depths and in challenging environments, though its primary focus has been polar regions. The limiting factor, the crude submersible built by Triton submarines and used in the Five Deeps expedition, is another. It is rated to full ocean depth, meaning it can theoretically descend to the deepest point in any ocean on Earth. And it has done exactly that, reaching the Challenger Deep in the Mariana Trench, the Malloy Deep in the Arctic Ocean, the Java Trench in the Indian Ocean, the South Sandwich Trench in the Southern Ocean, and the Puerto Rico Trench in the Atlantic. It is a genuinely remarkable machine, but it is privately owned, and access to it for scientific expeditions requires either purchase of ship time and vehicle time at significant cost, or collaborative arrangements that take time to negotiate and fund. The cost question is not a minor footnote. It is central to understanding why the Cayman Trough's deeper zones remain unexplored. A single day of research vessel time in the deep Caribbean, accounting for fuel, crew, port fees, and operational costs, typically runs somewhere between $50,000 and $100,000. A full expedition of 2 to 3 weeks, which is roughly the minimum time needed to transit to the site, conduct meaningful dives, and return, can cost anywhere from $1 to several million when you include the ship, the ROV or submersible, the science team, and all the associated logistics. That is before you account for the specialised equipment needed for full ocean depth operations, which adds further cost and complexity. Research funding bodies do fund deep-sea expeditions. The National Science Foundation, the Natural Environment Research Council in the UK, the European Research Council, and various national oceanographic agencies all support this kind of work. But funding is competitive, and the deep sea is a large place with many compelling targets. The Cayman Trough competes for attention and resources with the Mariana Trench, the Hadal zones of the Kermadec and Tonga trenches, the Mid-Atlantic Ridge, the Deep Arctic, the Deep Antarctic, and dozens of other sites that scientists have made strong cases for exploring. Being scientifically interesting is necessary but not sufficient. You also have to be the most compelling case in the room at a particular funding cycle, and the Cayman Trough has not always been that. There is also a practical constraint that is easy to overlook, which is the question of what happens when something goes wrong at extreme depth. Deep-sea ROVs and submersibles are expensive and difficult to repair in the field. If a vehicle sustains damage during a dive, either from an unexpected collision with the seafloor, a tether entanglement, a pressure seal failure, or a power system fault, the expedition may lose days or weeks of ship time while repairs are attempted. In the worst cases, a vehicle is lost entirely. The seafloor of the Cayman Trough, with its steep rift walls, unstable sediment and complex terrain near the spreading ridge, is not a gentle environment for vehicle operations. The risks are real, and expedition planners factor them into their decisions about where to send vehicles and how deep to push them. All of that said, the technological picture is not static. It is changing, and some of the changes are genuinely promising for future exploration of places like the Cayman Trough. Autonomous underwater vehicles, or AUVs, represent one avenue of progress. Unlike ROVs, which are tethered to a surface ship and controlled in real time by pilots, AUVs operate independently, following pre-programmed mission plans and navigating on their own. They can be deployed to survey large areas of seafloor, collecting sonar data, temperature and chemical measurements, and in some configurations, water samples, without requiring constant supervision from the surface. Because they do not have a tether, they can cover much more ground per deployment than an ROV. Some AUV designs are now being built to full ocean depth specifications, which would allow them to survey the Hadal zones of trenches like the Cayman Trough at relatively lower cost per square kilometre of seafloor mapped. The limitation of AUVs is that they are not well suited to biological sampling, at least not in their current form. Mapping the seafloor with sonar and measuring water chemistry is valuable, but finding and collecting biological specimens requires close-up imaging, precise manoeuvring, and the ability to respond to what you see in real time. An AUV flying 50 metres above the seafloor on a pre-programmed track will not stop to investigate a cluster of organisms it was not expecting to find. That kind of opportunistic observation still requires either a human in a submersible or a remotely operated vehicle with a skilled pilot watching the feed from the surface. So AUVs can fill in the mapping gaps, can tell us where the interesting terrain is, can identify areas of hydrothermal activity by measuring temperature and chemical anomalies in the water column, but they cannot by themselves answer the biological questions. Lander systems offer another approach. A lander is essentially a weighted frame carrying instruments and bait, dropped from the surface ship and allowed to sink freely to the seafloor. It sits there for a set period of time, attracting scavenging animals with the bait, recording whatever comes to investigate with cameras, and then releases its ballast weights and floats back to the surface for recovery. Landers are relatively inexpensive compared to ROV operations, and they can be built to full ocean depth specifications without the enormous engineering challenge of a crewed submersible. They've been used successfully in the Mariana Trench, the Kermadec Trench, and other Hadal environments to document the communities of scavenging amphipods, fish and other organisms that inhabit the deepest sea floor. A series of well-designed lander deployments in the Cayman Trough's Hadal zone could, at relatively modest cost, begin to answer the question of what lies at depth there. The fact that this has not been done is genuinely puzzling if you think about it for a moment. The technology is available, the cost is manageable, the scientific rationale is clear. And yet the Hadal floor of the Cayman Trough has never had a lander on it. The answer is probably a combination of factors. The Cayman Trough is not in the Pacific, where most Hadal biology research has been concentrated. It is not the deepest trench in its ocean, so it does not attract the attention of record-focused expeditions. It is not adjacent to a major oceanographic institution with a long-standing research program there. And the vent fields, which are the most obviously compelling targets in the trough, are at shallower depths that existing ROVs can reach. So the pressure to develop full ocean depth capability specifically for the Cayman Trough has been lower than it might otherwise be. The vents got the attention and the deeper zones got overlooked. Then there is environmental DNA, or eDNA, which is one of the more exciting methodological developments in marine biology over the past decade. The basic idea is straightforward. Every organism in the ocean sheds genetic material into the water around it, in the form of shed cells, mucus, feces, and other biological traces. If you filter a water sample and extract the DNA from whatever you catch in the filter, you can then sequence that DNA and compare it against known genetic databases to identify what organisms were present in that water, sometimes down to the species level, without ever seeing or catching the animals themselves. It is a bit like reading the guest list from the fingerprints left on a wine glass after the party is already over. eDNA sampling has been used in rivers, coastal waters, and increasingly in the deep ocean to detect the presence of species that are rare, shy, or simply hard to observe directly. In theory, a water sampling device capable of operating at Hadal depths, filtering large volumes of water and preserving the genetic material for later analysis, could be deployed in the Cayman Trough and recover information about what organisms are present in the water column and near the sea floor, even without physically capturing or photographing anything. The technology for deep eDNA sampling is still developing, and there are challenges around contamination, preservation, and the interpretation of results, but it represents a genuinely promising avenue for exploring biologically unknown environments. Applied to the Cayman Trough, eDNA sampling could potentially reveal the presence of organisms in the Hadal zone that no camera has ever seen. It would not give you images, it would not give you specimens to examine under a microscope, but it could tell you whether there are fish down there, whether there are crustaceans, whether the microbial communities are similar to or different from those at the vent fields. It could provide the first biological data from the deepest parts of the trough, and crucially, it could help scientists make the case for a more detailed follow-up expedition by demonstrating that there is something worth going back for. What is interesting, and perhaps a little frustrating, is that none of these tools are hypothetical. They exist. The landers exist. The AUVs exist. The eDNA methodology exists. Full ocean depth submersibles exist, even if they are rare. The Cayman Trough is not waiting for technology that has not been invented yet. It is waiting for the combination of funding, organisational will, and expedition planning that would bring existing tools to bear on a specific set of unanswered questions. That combination is not trivial to assemble, but it is not impossible either. It has been assembled for other trenches, other vent fields, other remote environments. The question is when it will be assembled for the deeper zones of the Cayman Trough. There is a broader point here about how exploration actually works in practice, which is worth saying plainly. The popular image of scientific exploration tends to involve dramatic expeditions to famous places, funded by major institutions, using cutting-edge technology, with results that make headlines. And those expeditions do happen, but a great deal of what remains unexplored in the deep ocean is unexplored not because the technology does not exist, and not because the science is unimportant, but because the specific combination of resources, timing, and institutional priority is not aligned. The Cayman Trough is a very good example of this. The scientific community knows it is interesting. The researchers who have worked there know it deserves more attention. The technology to explore it more deeply exists or is being developed. What has been missing is the alignment. That alignment could change. Funding priorities shift. New expeditions get planned. The 5 Deeps expedition demonstrated that private funding can accomplish things that traditional research funding has not prioritized. The growing interest in deep-sea biodiversity, partly driven by concerns about deep-sea mining and its potential impacts on unknown ecosystems, has increased attention to Hadal environments generally. The Cayman Trough sits in a part of the ocean that is increasingly relevant to these conversations, given the mineral-rich seafloor associated with its spreading ridge. There are, in other words, multiple reasons why the next decade might bring more sustained attention to the trough than the previous one has. And when that attention arrives, when the landers go down, when the AUVs map the Hadal floor, when the eDNA filters are retrieved and the sequences are run, what they find will matter in ways that extend well beyond the Cayman Trough itself. Because what the trough represents, in its combination of extreme depth, active geology, biological isolation, and almost complete lack of biological data from its deepest zones, is precisely the kind of environment where the boundaries of what we know about life on Earth are most likely to be pushed outward. The tools are ready. The questions are clear, and the creatures, whatever they are, have been going about their business in the dark for a very long time, entirely indifferent to the question of whether anyone is coming to look. What those creatures might actually tell us, both about life in this trench and about the larger question of what life can be, is where this story goes next. So let us sit with that question for a moment. What would it actually mean? in a real and concrete sense, to find genuinely new species in the Cayman Trough. Not just new to the trough, but new to science entirely. Organisms with body plans not yet catalogued, metabolic strategies not yet described, behaviours no researcher has ever watched. It is easy to say that such a discovery would be significant and then move on. But the significance runs in several different directions at once, and each of them is worth following a little way. The most immediate implication is biological. The deep ocean already holds some of the strangest life on Earth, and the Cayman Trough's combination of extreme pressure, chemical energy from the vents, and long isolation creates conditions where evolution has had both the raw material and the time to do unusual things. Every new species found in an environment like this adds to our understanding of what life can tolerate, what it can build with, and how far it can diverge from the forms we consider familiar. The shrimp Rimicaris hybaceae, discovered at Beebe in 2012, was not just a curiosity. It was evidence that the vent communities of the Cayman Trough had their own evolutionary trajectory, distinct from their relatives at shallower Atlantic vents. Its adaptations to depth and pressure told researchers something about how organisms respond to extreme conditions over generations. A new species from the hadal floor of the trough, from the zones below 6,000 metres that have never been sampled, would likely be even more divergent. It might represent an evolutionary lineage that has been separated from its nearest relatives for millions of years, shaped entirely by conditions that no surface organism ever experiences. There is also the question of what new species from the trough might offer in a more practical sense. This is not the most poetic way to think about deep-sea discovery, but it is a real one, and it deserves honest attention. Extremophile organisms, meaning organisms that thrive in conditions most life cannot survive, have already contributed directly to human medicine and biotechnology. The most famous example is probably Taq polymerase, the enzyme that makes the polymerase chain reaction possible. PCR is the technique used to amplify DNA for genetic testing, forensic analysis, medical diagnostics, and research of almost every kind. The enzyme that makes it work was isolated from Thermus aquaticus, a bacterium discovered in the hot springs of Yellowstone in 1966 by Thomas Brock and Hudson Freeze. That single discovery from a geothermal environment, a discovery that at the time had no obvious commercial application, eventually became foundational to modern biology. The biotechnology industry built around PCR is worth tens of billions of dollars, and it started with a bacterium in a hot spring. The organisms living at the Beebe vent field, at temperatures exceeding 400 degrees Celsius at the vent source, and in water saturated with minerals that would be toxic to most life, have evolved enzymes and biochemical processes that function under conditions that would destroy the proteins of a surface organism. Those enzymes are potentially interesting for the same reasons that Taq polymerase was interesting. They do things that ordinary biological molecules cannot do. They remain stable at high temperatures, or high pressures, or in the presence of chemicals that would denature a normal protein. Whether any specific compound from a Cayman Trough organism would turn out to be medically or industrially useful is impossible to predict. That is the nature of basic research. You do not know what you will find until you look, and you do not know what it will be useful for until much later. But the track record of deep-sea and hydrothermal organisms producing biochemically novel compounds is strong enough that the potential is real, not just rhetorical. Beyond the practical, there is a dimension to this story that reaches further out than the Caribbean, further out even than Earth. The hydrothermal vents of the Cayman Trough have become, over the past decade or so, a reference point in the field of astrobiology. That might sound like a stretch at first. The Cayman Trough is in the Caribbean. Astrobiology is about life in space, but the connection is direct and the reasoning is solid. The fundamental challenge of astrobiology is figuring out where in the solar system life might exist, given what we know about the conditions life requires. For a long time, the working assumption was that life needed liquid water, a source of energy and organic chemistry, but that the energy source had to be at some level connected to sunlight. Photosynthesis drives almost every ecosystem on Earth's surface. Even ecosystems that seem to have no direct connection to sunlight, a cave system, say, or a deep soil community, typically depend on organic matter that was produced at some point by photosynthetic organisms. The food chain leads back to the sun. Hydrothermal vent ecosystems broke that assumption. The organisms at the BB vent field do not depend on sunlight, not even indirectly. Their energy comes entirely from chemical reactions involving the minerals and gases in the vent fluid. The bacteria at the base of the food web oxidize hydrogen sulfide and other compounds to produce the energy they need to fix carbon, building organic molecules from scratch without any input from the surface. Everything above them in the food chain, the shrimp, the crabs, the fish, the worms, is ultimately sustained by that chemical energy. The sun is irrelevant. It could be permanently extinguished and the vent ecosystem would continue, at least until the geological processes driving the vents eventually changed. This matters enormously for astrobiology because several bodies in the outer solar system are now believed to harbour liquid water oceans beneath their icy surfaces, sustained not by solar warmth, but by tidal heating from the gravitational pull of their parent planets. Europa, the moon of Jupiter, has a subsurface ocean that may be in contact with a rocky seafloor. Enceladus, the moon of Saturn, has an ocean that is actively venting through its surface ice, and the plumes it produces have been sampled by the Cassini spacecraft and found to contain water vapour, ice particles, organic compounds, and molecular hydrogen, which is exactly the kind of chemical signature you might expect from hydrothermal activity on the ocean floor. Both of these moons are now considered among the most promising places in the solar system to look for life. And the reason scientists think life might be possible there is precisely because of what has been found at places like the BB vent field. The logic runs like this. If life can exist at nearly 5,000 meters depth in the Caribbean, in total darkness, at crushing pressure, sustained entirely by chemical energy from the Earth's interior, then there is no fundamental reason why life could not exist in the subsurface ocean of Europa or Enceladus under broadly similar conditions. The Cayman Trough is in this sense a proof of concept. It demonstrates that the conditions we once thought were necessary for life, specifically sunlight and a connection to the surface world, are not actually necessary. Life is more flexible than that. It finds a way to extract energy from whatever chemistry is available, and it builds ecosystems from there. This does not mean there is life on Europa. It means the possibility is real in a way that it would not be if the only life we knew of was photosynthesis dependent. The Cayman Trough vents and the organisms living there have expanded the definition of a habitable environment. They have made the universe a more plausible place for life to exist. That is not a small thing. The deeper zones of the trough, the Hadal floor below 6,000 meters, add another layer to this picture. If organisms can survive not just at vent temperatures and pressures, but at the extreme conditions of the Hadal zone, combining crushing pressure with near-freezing temperatures and complete darkness, then the envelope of what counts as habitable expands further still. We already know from the Mariana Trench that Hadal fish like Pseudoliparus sweri can thrive at depths exceeding 8,000 meters. We know that amphipods swarm the deepest trenches in large numbers. We know that microbial communities exist in the sediment at Hadal depths, metabolizing organic matter that drifts down from the surface. If the Cayman Trough's Hadal zone holds similar communities, or different ones adapted to the trough's specific geology and chemistry, that adds another data point to the picture of what life can do under pressure. There is also something worth saying about what it means that the Cayman Trough exists where it does. Not in the remote Pacific, not in the polar regions, not in some corner of the ocean that requires months of transit to reach. The trough lies in the Western Caribbean, in a sea that is one of the most travelled and most commercially active bodies of water on the planet. Cruise ships cross it, cargo vessels cross it, tankers cross it, fishing fleets work its surface, tourist flights pass over it daily. The ports of Jamaica, Cuba, the Cayman Islands, and Central America frame its edges. It is, in the most literal sense, a familiar sea. People vacation above it. People have vacationed above it for generations, and yet, kilometres below the surface of that familiar sea, in the cold and the dark, there are creatures that no human being has ever seen. Not because they are hiding, not because they are rare in the way that large charismatic megafauna can be rare, but simply because no one has gone deep enough, in the right places, with the right tools to find them. They are there, in the sense that the biology of the trough and the history of every other hadal environment ever studied makes their presence not just plausible, but expected. They are there, doing whatever it is they do, feeding, moving, reproducing, dying, contributing to ecosystems that function on timescales and in conditions that have nothing to do with the world above. There is something quietly humbling about that. We tend to think of the unknown as distant, as something that requires enormous effort and expense to reach, as something that lives in the far corners of the map. And sometimes that is true. The surface of Mars is genuinely far. The interior of Europa is genuinely inaccessible with current technology. But the unknown is also sometimes just below. It is in the water beneath the sea we already know. It is in the dark below the reefs and the shipping lanes and the tourist beaches. The Cayman Trough is not an exotic frontier in the way that a distant planet is exotic. It is close, it is right there, and it is still, in its deepest parts, completely unknown to us. That proximity makes the situation feel different than it might otherwise. There is something almost startling about the idea that a creature, perhaps a fish, adapted to 7,000 meters of pressure, perhaps a crustacean with biology shaped by millions of years of isolation, perhaps a microbial community with chemistry we have never described, is alive right now, in water that is geographically a short distance from some of the most visited resort beaches on Earth. The distance is not geographical, it is vertical. And vertical distance in the deep ocean turns out to be one of the most effective barriers to human knowledge that exists. It is also worth sitting with the fact that these creatures, whatever they are, do not need to be found. They are not waiting for discovery in any meaningful sense. The trough existed for millions of years before any human being knew it was there. The vents at Beebe were producing their plumes, and the shrimp were swarming their chimneys for a very long time before the RRS James Cook arrived in 2010. The Hadal floor has been accumulating sediment, and whatever lives in it has been living in it, entirely without reference to whether a research vessel is overhead or not. Discovery is a human act. The thing being discovered has its own existence that predates and does not depend on being known. That might sound obvious, but it is worth saying because it changes the emotional register of the question slightly. The Cayman Trough is not a mystery in the sense of something waiting to be solved. It is simply a place that exists and that we have not yet looked at carefully. The creatures in it are not mysterious in any mystical sense. They are biological organisms following the logic of their own evolution and their own environment. What makes them interesting to us is not that they are strange in some cosmic way, but that they are unknown, and that the process of making them known would tell us things we currently do not know about life, about chemistry, about the limits of what biology can do. And that process, when it eventually happens in a more sustained way than it has so far, will be careful, slow, expensive, and probably not very dramatic to watch from the outside. Researchers will spend months planning expeditions, writing grant applications, calibrating instruments. Ships will transit to the Caribbean. ROVs will be lowered into the water. Landers will descend on long cables. Samples will be collected, preserved, and brought back to laboratories. DNA will be sequenced. Morphology will be described. Papers will be written and peer-reviewed and published in journals that most people will never read. The process is not cinematic. It does not look like adventure in the popular sense. But what it produces, if it is done carefully and thoroughly, is knowledge. Real knowledge. The kind that changes what we know about life on Earth, and perhaps about life elsewhere. The kind that occasionally produces something as unexpected and consequential as an enzyme from a hot spring that ends up changing the entire field of molecular biology. The kind that shifts the boundaries of what we consider possible, quietly and permanently. The Cayman Trough is ready for that kind of attention. It has been ready for a long time. The biology is there, waiting in the dark, going about its business in the cold. The geology is active, producing the chemical energy that sustains it. The isolation has been doing its work for millions of years, shaping whatever lives there into forms that may be genuinely unlike anything we have catalogued. All that is needed is the sustained, systematic effort to go and look. What that looking might reveal, and what it might tell us about the nature of life itself, is something that no one can fully predict. That uncertainty is not a weakness in the story. It is the point. The Cayman Trough holds questions that have not yet been asked properly, because the tools to ask them have only recently reached the place where asking them is even possible. The next expedition that goes deep enough, that samples the right water, that deploys the right instruments in the right places, may come back with answers that reshape the conversation entirely. Or it may come back with new questions, which in science is often the more valuable outcome. Either way, the trough will be there, patient and cold and entirely indifferent to the timeline, holding whatever it holds in the dark below the warm Caribbean Sea. And the story of what lives there is still, in every meaningful sense, waiting to be told. So let us come back just for a moment to the surface. Picture it the way it actually looks. Turquoise water catching the afternoon sun. A gentle swell moving through it, unhurried. Somewhere nearby, a boat is anchored, and someone is putting on fins, about to slip into the warm shallows above a reef. The water is clear enough to see the bottom from up here. The fish are colourful and familiar. The whole scene is bright and warm and completely legible to the human eye. This is the Caribbean that people know. This is the version that exists on travel posters and in the memories of anyone who has ever been here. And directly below it, kilometres below it, something else entirely is happening. The water cools as you descend, not gradually in a way that feels like a transition, but in stages that become increasingly absolute. Below 1,000 metres there is no sunlight at all. Not dim light, not filtered light. No light. The water is not dark in the way a room is dark when you turn off the lamp. It is dark in a way that has no reference point in ordinary human experience. It is the kind of dark that has simply never been interrupted by anything luminous, because nothing luminous has ever been there. The temperature near the seafloor, away from the vents, sits somewhere between 2 and 4 degrees Celsius. The pressure at the deepest point of the trough, at around 7,686 metres, is approximately 770 atmospheres. That is a number that is genuinely difficult to make feel real. At that pressure, the weight of the water column above is equivalent to roughly 11,300 pounds, pressing on every square inch of whatever exists down there. For a biological organism, that kind of pressure would be instantly lethal without very specific adaptations. For the organisms that live there, it is simply the normal condition of existence. They have never known anything else. And somewhere in that cold, pressurized, lightless water, things are moving. We know some of them. The BB vent field, sitting at around 4,960 metres depth, has shrimp swarming in their thousands across the surfaces of chimney structures that reach temperatures above 400 degrees Celsius. Rimicaris hybaceae, the species described in 2012, clustering in dense mats where the chemistry is right, where the bacteria they depend on are growing in the mineral-rich fluid that pours from the seafloor. The Von Dam vent field, higher up on the flank of an underwater mountain at around 2,300 metres, has its own community, including the Hoff crab, that peculiar relative of the Yeti crab with its hairy chest, adapted to cultivate chemosynthetic bacteria. moving slowly through the diffuse warmth of lower temperature vent fluid. Chemosynthetic bacteria forming the invisible foundation of all of it, pulling energy from hydrogen sulfide and methane, building the base of a food web that has no connection whatsoever to the sun shining on the surface above. These are not hypothetical creatures. They were collected, photographed, described, and published in peer-reviewed journals. They are as real and documented as any organism in a museum collection. The fact that they live in conditions that would kill a human being in seconds does not make them less real. If anything, it makes them more remarkable, because they are evidence of something important. They are evidence that life, given the right chemistry and enough time, will find a way to work in almost any environment the Earth can produce. But the BB vents and the Von Dam vents represent a small and specific portion of the trough. They are the places scientists went on expeditions that were expensive to mount and limited in time. The RRS James Cook arrived in 2010 and made history simply by getting there, by lowering instruments and cameras to a depth that had never been visited with that level of scientific intention. The follow-up expedition in 2013 returned to the same vent fields, collected more specimens, filmed more of the community, and still left most of the trough untouched. The sediment plains between the vents were not sampled. The walls of the rift were not surveyed for biology. The deeper trench axis, below 5,000 metres, was not systematically explored for life, and the Hadal zone, the deepest portion of the trough, below 6,000 metres and reaching all the way to 7,686 metres at its maximum, has never had a biological lander or any crewed or uncrewed vehicle conduct a proper survey of what lives there. That is not a gap in our knowledge. That is an absence of knowledge. There is a difference. A gap implies that we have some data on either side and are missing the middle. An absence means we simply have not looked. The deepest part of the Cayman Trough is, in terms of biological documentation, essentially a blank. What might be there is genuinely unknown, and it is worth being careful about the distinction between what is hypothesized and what is confirmed. Scientists who study Hadal environments have reasonable grounds to expect certain things. Hadal-adapted fish are known from other deep trenches. The Mariana Trench has Pseudoliparis swirei, the Mariana snailfish, documented at depths around 8,000 metres. Amphipods, those small crustacean scavengers, have been found in enormous numbers in Hadal zones around the world. Microbial communities in the sediment of deep trenches are now known to be diverse and metabolically active. There is no particular reason to think the Cayman Trough's deepest zones would be lifeless. The chemistry is there. The organic material that drifts down from the surface world provides a food source for scavengers. The sediment itself is a habitat for microbes. But none of that has been confirmed for the Cayman Trough's deepest zones. It is reasonable inference, not documented fact, and the history of deep-sea biology is full of cases where reasonable inference turned out to be incomplete, or wrong, or simply insufficient to prepare researchers for what they actually found. The BB vents themselves were not predicted in detail before they were visited. The specific species found there, the density of the shrimp swarms, the particular chemistry of the vent fluid, these were not known in advance. They were discovered. And discovery, by its nature, involves encountering things that were not anticipated. The Cayman Trough has been in its current geological configuration for a very long time. The Mid-Cayman Spreading Centre has been producing new seafloor at its slow rate of roughly 15 millimetres per year, across a timescale of millions of years. The isolation of the vent communities from other Atlantic vent systems has been ongoing for a comparable period. Whatever has been evolving in the deeper, unvisited zones of the trough has had that entire span of time to diverge from its nearest relatives, to adapt to its specific conditions, to develop whatever biological strategies allow it to survive in that particular environment. Genetic divergence takes time, but given enough time, it produces organisms that are genuinely distinct. Rimicaris hybaceae already shows this. The deeper zones, with their greater isolation and more extreme conditions, may show it even more dramatically. There is also the question of the vent transition zones, the areas at the edges of vent influence where hot fluid mixes with cold ambient water. These gradient environments, where chemistry and temperature shift over short distances, are known in other vent systems to host their own distinct communities. Organisms that are neither fully vent adapted nor fully cold water adapted, but occupy the boundary between the two. At the Cayman vents, some of these transition zones were observed during the 2013 expedition but not fully catalogued. Organisms were photographed that were not collected. The equipment constraints of the expedition meant that some things were seen and then left behind. They are somewhere down there still, going about whatever they do, unnamed and undescribed. It is a strange kind of situation to sit with. We live in a period when it is genuinely possible to know in real time what is happening on the surface of Mars. Rovers send images, satellites map the terrain, the atmospheric chemistry of a planet 225 million kilometers away is being measured and analysed, and simultaneously, a trench in the Caribbean Sea, a body of water that lies between islands where millions of people live and vacation and work, contains zones that no instrument has ever reached and no camera has ever filmed. The contrast is not a criticism of anyone's priorities. It is simply an observation about the particular strangeness of our moment in history and about how the distribution of human attention and human resources does not always follow the distribution of what is unknown. The tools to go deeper exist or are being developed. Full ocean depth vehicles have been built. Environmental DNA sampling offers a way to detect species presence without physical collection. Autonomous underwater vehicles are becoming more capable and less expensive. The scientific community that studies deep-sea biology is aware of the Cayman Trough and its significance. The knowledge is there, the motivation is there. The question is whether the sustained funding and institutional attention will arrive in time to make the next round of expeditions meaningfully more comprehensive than the last. That is not a question this narration can answer. It is a question that belongs to the future, to the researchers who will write the grant applications and plan the ship time and lower the instruments into the dark water. What this narration can do is sit with the fact of what exists down there, known and unknown together, and let that fact be what it is. The Cayman Trough is not waiting for us. That is perhaps the most important thing to carry away from this. It is not a story with a cliffhanger, not a mystery and suspense, not something that exists in a state of anticipation. The shrimp at the BB vents are swarming right now, in this moment, whether or not anyone is watching. The spreading centre is producing new seafloor at its slow, patient rate. The hydrothermal plumes are rising into the cold water column. The sediment is accumulating on the Hadal floor. Whatever lives in the deepest zones is living there, metabolizing, moving, reproducing, dying, in total darkness and total silence, in a world that has never required human observation to function. This has been true for millions of years. It will continue to be true whether or not the next expedition happens next year or in 50 years. The trough is not fragile in that sense. It is not waiting to be saved or waiting to be discovered. It simply is. What changes when we look is not the trough. What changes is us. Our knowledge expands. Our picture of what life can do becomes more complete. Our understanding of the chemistry of the deep sea floor becomes more precise. Our models of how isolated ecosystems evolve become better calibrated. And perhaps, in ways that are difficult to predict in advance, our understanding of where else life might exist in the universe becomes a little more grounded. The Caribbean above the trough will continue to be what it is. The warm water, the reefs, the tourist boats, the cargo ships crossing from one port to another. None of that is diminished by the knowledge that kilometres below, in conditions those surface travellers will never experience, something genuinely unknown is alive and moving. If anything, it adds something. It adds a layer to the world that most people never think about, a depth that is literal as well as figurative. And for those of us who do think about it, even briefly, even just for the length of a quiet evening spent listening to someone describe it, There is something in that thought that is worth holding on to. The world is larger than it looks. The unknown is closer than we assume, and the creatures that live in the dark below the warm sea do not need our attention to be extraordinary. They already are. Thank you for spending this night here, for following the trough from its surface all the way down through the geology and the vents and the shrimp swarms and the Hadal silence, for sitting with the parts of the story that do not resolve neatly, because they genuinely have not resolved yet. That kind of open-ended wondering is, in its own way, the most honest thing science offers. Not all the answers, but a clearer picture of where the questions live. Wherever you are listening from tonight, whatever the hour is, whatever the temperature outside your window, I hope this was a good place to spend a little time. Sleep well, rest easy, and know that somewhere in the dark below the Caribbean, the trough is doing exactly what it has always done. Patient and cold and full of life we have not yet had the chance to meet.